Okay, we're live. Hey, it is Friday. It is the December 2nd, 2016. I have 3 o'clock Pacific time, and I've got a couple people joining us today. Ben Kissler. Is that Kissling? Is that how you pronounce his last name?
Kissling, yeah,
Kissling.
it's a lot of people get it wrong. They say Kisslinger a lot, but
It's just
pretty
Kissling, right?
easy.
Ben
Yeah,
Kissling?
Kissling.
Okay. Ben Kissling, he is a member of the Creationism forum on Facebook. He is a younger creationist, and he's kind of going to be walking us through some of Tomkins' claims directly from Tomkins' work that he's done on human chromosome number 2. He has actually talked to Tomkins, and he has said that Tomkins may actually join us in January, depending on how this goes. So, we're gonna be treating him with the utmost respect and allowing Ben to walk us through this. And basically, we're going to be asking him questions while he does. We also got CRISPR here to join us to kind of to guide us as well. Other people I've invited have been Rational Mind, Isaiah Sloan, and Dr. Herman Mays, who's a zoologist. So they may join us. If people want to join in the live feed, let me know, and I will give you the link privately, and we'll go from there. But anyways, Ben, introduce yourself.
Hey, I'm Ben Kissling, and I grew up in your typical fundamentalist Christian evil home where everyone is indoctrinated in the Bible. And basically, I started getting interested in science, specifically biochemistry, when I read Darwin's Black Box in, I believe it was a junior in high school. And because of that book, I studied biochemistry in college, and I intended to become a research scientist, but I kind of lost interest in that. Basically, now I work in a lab as a lab tech. I'm a lowly lab tech. I've been called that. So, but it's true. I was just a lab rat. Actually, I recently switched positions. I'm in the records now, which is a little easier. But...
Correct me if I'm wrong, you have a bachelor's degree in biochem, correct?
Oh, yeah.
Okay.
Sorry, at the University of Nebraska, Lincoln is where I went. And I grew up in Lincoln, Nebraska. And I love the Cornhuskers, which is actually a big part of the reason why I didn't come last week, is because the Cornhuskers were playing at 2 o'clock.
Got to support your team, right?
Right, 2 o'clock on Friday was their game, so I was like, I think I'd rather watch that. But I shouldn't have. We got smashed.
Did they? Oh, that's a shame. I don't follow that much with sports. I got to admit, I did watch the last game of the World Series. It's a World Series, right? And the Cubs winning is this, first time in like what 100 years or something like that.
Yeah, it's a big deal.
Crazy. And then I'll watch the Super Bowl. But other than that, just not much into sports.
Oh, and also one more thing. I'm currently living in Dallas, so I am actually.
Hey, you're in our friend Tommy Hall's world. Isn't he from Dallas? Anybody know? He's another younger creationist.
Okay, but maybe I might actually be able to get to score a meeting with Tomkins sometime. We'll see. I've never actually been to ICR, but I've driven by it. So.
Well, we have reached out to him before, as I've told you before. I've talked to you a couple times in the forum. Ruhif, was the one that actually had talked to Tomkins and explained to him that his 70% genomic analysis was flawed because of a bug program blast, and Tomkins did realize that. I don't know what the situation between them is. What's that?
He published a paper on it last year, and
he retracted it. Yeah. Well, I don't think it was a published paper, but he wrote something on it, and then he retracted that because there was a bug program blast.
He has a published paper on it.
Was it?
It's in ARJ. Yeah, he basically tested all the different versions of BLAST and found out that the oldest one was the only one that did not have the bug, and reran the simulation several times and came up with 88%.
Right.
He also ran it with another couple programs, one called NuckMare, which correlated with 88%, and then another one called LastZ, which gave 73%, which he actually didn't trust as much. So.
The ones that he ran though later on with the blast, I mean, he didn't use an ungapped parameter, and that allowed him to use indels and treat the indels as multiple different types of genomic differences rather than one specific difference.
I am not sure about that. All I know is that he removed the ends from the sequence, and he has reasons for that, which are in his 2011 paper, which I'll probably talk more about later.
Okay.
After talking with him, I don't think that he. I think that he would say that the published chimpanzee genome is flawed and that it's not trustworthy. And he gives more of why he thinks that actually in the 2011 paper, and we can go over that if you'd like.
Okay, well, we're gonna start off the human chromosome number 2 paper for. Or I hate to call it a paper because if it's published in NIG, I can't call it a published paper.
And that's part of the thing where I say you need to be treating him with more respect. Because if you just wave your hands and say his work that he did and his reference, just like everybody else, is not worth looking at and is not worth respecting as much as anybody else, that's his work. You're having him on.
Right. We're going to call it his work.
On to discuss that.
Well, that's what we're going to be discussing. That's why we're discussing it. I just can't call it a published paper because it technically
it is published, and it is a paper. It is a published, peer-reviewed paper.
What peer-reviewed journal was it in? Do you know?
ARJ.
That's not a peer-reviewed journal.
Yes, it is a peer-reviewed journal. He's recommending peer reviewers for it right now for publishing a new paper.
Christopher, you want to weigh on that?
We're not here to talk about what constitutes a published paper. Let's talk about human chromosome 2.
Okay, so we're gonna. I'm gonna put the actual link into the live feed if people want to follow along. I'm going to put the one from AIG. It's called Alleged Human Chromosome Number 2 Fusion Site, encodes a active DNA binding domain inside a complex and highly expressed gene and getting fusion. Is that the one you want to use?
Alleged Human Chromosome 2 Fusion Site encodes an active DNA binding domain inside a complex and highly expressed gene.
Okay, that's the one I'm using
fusion
as well.
Is the one I have.
Okay, right. So once you start us through that, and we'll go through like each of the claims as you're going through that. Because go ahead and tell us what Tomkins is claiming with this, and then we'll go from there. Sound good?
Okay. Well, the basic argument is that obviously humans and chimpanzees have different numbers of chromosomes. It's just to set everything up. I'm sure most people already know this. But humans have 46 chromosomes, 23 pairs, and chimps have 48 chromosomes or 24 pairs. So if they have a common ancestor, you have to explain from an evolutionary perspective why there's a different number of chromosomes. And the way evolutionists do that is they say there's a fusion between chimp chromosome 2A and chimp chromosome 2B. And they say they have a specific site where they believe the chromosome fusion occurred. Now, the site is a 798 base pair region that is a telomeric repeat. So a telomere is a sequence of repetitive DNA at the end of chromosomes. When I was in college, there was some ridiculous stuff going around about telomerase, a protein that extends telomeres. And the reason that's important is because when DNA replicates, it loses a short segment of each end every time it replicates. And so after a while, you'll run out of telomere and you'll start losing useful information that you need. And so the idea was telomerase, if we could get it to activate, would basically make cell lines immortal. Well, we know now which kind of cell lines are immortal, and those are cancer cells. And that's a lot of the cell lines that are used in the ENCODE project is cancer cells because they're immortal and they don't run out of telomeres. Tomkins told me there was actually a cancer cell from a 1950s woman who had cervical cancer that's still being used.
Henrietta Lacks, I believe her name was.
Anyway, so that's a telomere, and so the idea is that we
have some of those cells in our lab, so.
The HeLa. They call that. Right, they show them called HeLa, I believe.
I'm sure this is not news to you, CRISPR. I'm just going through it. So the first thing here is a diagram where he shows the matching of the chimp chromosome 2A and chimp chromosome 2B, where the centromeres are. So a key aspect of this is that the human chromosome has one centromere that actually works, and what they think is a cryptic centromere that is the non-working, non-functional leftover centromere. So that's part of the claim as well.
Now, Tomkins doesn't deny there's a cryptic centromere, correct?
Well, he doesn't believe that it's an actual centromere from the chimp chromosome, if that's what I mean. He has a series of arguments based on alphoid sequences that that he argues that that's not
That the
actual,
alpha sequence there is not a cryptic centromere.
that the alphoid sequences are found all over the genome.
Just a quick thing. While we're on the subject of that figure, could you take a moment to explain how it is exactly that he drew that figure?
Let's see. I have what he said on that.
Yeah, and just I have sent you all Ruhif's information on this document.
Yeah, and
Ruhif, so I want people to know that we're not sharpshooting anybody. This has already been
I'm actually going to be discussing this in Ruhif's terms, in terms of his critique. So Ruhif says that this figure is not accurate because it doesn't match, the published data or whatever. Now, hold on. Okay.
No problem. While you're doing that, I want to welcome I'm going to get this right. Erman Mays. I haven't spoken to you, actually in person, Erman. So this is the first time. Yes, I can hear you.
Oh yeah, it's Herman.
Herman. Okay. Herman is also from the Creationist group. Very nice gentleman that I met there. He is a. Can I say your background?
Yeah, go ahead.
He is a PhD in zoology. I believe you teach at Maryland. Is that the?
I teach at Marshall University in West Virginia.
West Virginia. Okay.
The position I was hired in is a genetics position, so I teach genetics, undergraduate genetics, and graduate courses in population genetics.
And you, of course, know Ben.
I know Ben.
Okay.
From the Creationism group. Yeah.
Wait, who is this?
It's Herman.
Herman. Oh, you said Herman, and I didn't recognize it.
Well, what's funny is we have something in our group. Her name's Hermine, and she has it with an H, and everybody keeps calling her Hermine, and it's Hermine. So I wasn't sure if he was German, or something.
People call her.
I read something recently that said that the average age of Herman nowadays is like 86 or something. So it's not.
That name hasn't been around for a while.
It's been. The problem is it has been around for a while, but not too many people are naming their kids Herman.
Well, that's what I'm saying. It hasn't been utilized in a while. It hasn't been. Yeah.
Right.
They're all like old farts.
Yeah, it's old art. Yeah, I'm just old-ish art.
Well, welcome. Glad you can join us.
Sure.
Herman and I have actually talked a lot on the forum. I actually kind of like Herman, so
he's a nice guy.
I'm happy you're here. All right, so I posted in the comments the reference that Tomkins was using to draw his picture.
But to be fair, they don't actually in that reference they don't have the sizes. It's somewhat difficult to say exactly how Tomkins came up with this because he doesn't have a method section in this paper. But based on the text associated with that figure, it appears that he eyeballed the sizes in a 30-some-year-old paper and then drew it and then measured that and then said that the difference between the two things he drew based on an eyeballed 30-year-old micrograph was 10%.
Okay, so this is actually going to speak to a lot of the reasons why he doesn't trust the published Chimp genome. And so to go through this, we're going to have to, again, I'm sorry if this is not news. I know it's not news to you.
No, it's fine. It's fine.
So when the Human Genome Project was begun in 1990, they started with a genetic map, and that's a linkage map, different from a sequence. So genome maps and linkages are different from sequencing. You have to have a genetic map, which is based on originally restriction enzymes, I believe, and now they're based on markers. This is work that I actually have done in the lab. So you have to have this map to paste your sequences onto, basically, because or else you don't know where all these fragments are in the genome. You can sequence them fine, but you have to put them all together. And so genome sequencing in that method, the older method, is more rigorous and it's more accurate than the technique that is prevalent now called shotgun sequencing. So what happened was the Human Genome Project was going through this rigorous old method of using linkage map to map their data to the genome. And a guy named Craig Venter came along, who I actually like. I think he's kind of cool, but he came along and said, well, I can do this a whole lot faster than they can. And he did it in the private sector with a company called Celera. And he's still doing his work in the private sector, but I think. But he came along and used this whole genome shotgun gun technique to basically, it was a lot faster basically. But the problem with it is that you had to have that linkage map to paste everything together basically. And that's really, I could be butchering that, but essentially he used the original data from the Genome Project that they published to paste all the information on there. Now the relevance to the chimp genome is that nobody has actually done the original gene linkage map for chimpanzees. What they did with the chimp genome was they used their evolutionary assumptions that the chimp genome is going to be very similar to the human one, and used the human genome as the linkage map, as the map to use the shotgun sequencing data and paste it on there. So that is why Tomkins does not trust the published chimpanzee genome. It's not because of some numbers that are in BLAST or things like that. It's because he knows the wet chemistry that went behind it, and he knows that the evolutionary assumptions are baked into the chimpanzee's genome from the start. So it's not surprising that this data gets aligned almost perfectly. It's because it's intentionally aligned perfectly in order to sequence it from the beginning. So, and the reason he's accessing these old papers are because these old papers, they actually did the hard work of the linkage mapping and cytological chemistry, wet bench chemistry experiments that they use to determine things like genome size. And we know from experiments like that, and I can post a paper like this as well, that shows that the chimp genome is 8% larger than the human genome. So right there is an 8% difference. And we know that's true. It's so
I'm sorry though, but so my question was, how did he draw this figure?
He determined that there was a 10% loss in the information from the I believe the subtelomeric region from another study. Sorry, I'm looking through the thing here.
Okay, so what he says is, all chromosomes were comparatively drawn to scale according to cytogenic images by Eunice and Prakash in 1982. Okay, so what he did...
That's not what I said.
That's what he said.
No, there's other things that are going into his reasoning, and I'm not quite...
Well, then he should have included it in the paper.
Fiction of a hypothetical scenario. Yeah, I'm not really sure about the reasoning there. All I know is that he thinks there is a significant loss of DNA at that portion, and with other methods of
doing this,
for instance, this is a subtelomere region, correct? The fusion site.
The thing is, so what I'm trying to say is that he is basing this 10% loss on comparing the micrographs of these two stained chromosomes, or three technically, stained chromosomes, and he's, I guess, I don't know, measuring them with a ruler held up to the picture, and then he's drawing them based on that.
He says approximately. Is that not good enough for you?
Well, correct me if I'm
No,
wrong. I'm trying to
because-
find Ruhif's thing. When Ruhif did the measurements and used actually the actual base pairs, he came out with a completely different little picture that was a lot more aligned, right?
Steve, I
And
understand.
that's what I was talking about before. Is that of course it would be aligned because it's aligned purposely to help sequence the chimp.
And Ben, let me say that I have no problem with you rejecting it on that basis if that's what you want to do. What I'm saying is that his methodology for measuring these is completely flawed. Okay, you can't take a
So maybe you can
30-year-old micrograph.
It doesn't
right?
matter how old it is. Is the data valid or not? Are you questioning the basis of the paper?
Is the
Or
basis of the paper? Is the
maybe
basis?
maybe you can explain to me why you don't think that paper had accurate information in it. I'm willing to listen.
I'm sorry. You mean why are you saying why I don't think that the Unison Prakash paper had accurate information?
Yeah, that's the paper we're talking about, right?
Well, we're talking about Tomkins' paper, but the Yunus and Prakash paper has accurate information if you use it appropriately. You
So
can't.
explain to me. I have not read that paper, so you'll have to explain to me why you think that he did not use that correctly.
Because you're talking about micrographs of gem-sustained chromosomes. You don't just measure that and say, well, now I know what the base pair length is.
Well, from my understanding, that's exactly the kind of method that was used to propose a fusion on human chromosome 2 in the first place, was it not? Was banding?
It's that the banding patterns align with one another. That doesn't have anything to do with the
The
sequence.
size.
Yeah, people were able to match up banding patterns between different
And
chromatids.
what is a banding pattern but a measure of size of?
No, the
These are polymer. These are
No,
electrophoresis, are they not?
No, not
No.
electrophoresis.
patterns are stains that correspond to different types of chromatin in a chromosome. So those bands you line up, there's no base pair measure associated with those. They're just a pattern of bands that you can line up between two chromosomes. This was done way before we could ever.
This was like in the 80s, wasn't it?
Yeah, this was before we could think about sequencing these chromosomes. Nobody had sequenced them, but we could.
Well, no one had sequenced a chromosome, but they'd sequence things like small proteins and things like that.
Of course, but in this data set, they're talking about they're not measuring base pair size things. They're talking about lining up two different chromosomes from two different species that have similar banding patterns based on these stains that are banding.
But that's what the diagram is lining up chromosomes from two different species according to sequences that are similar on each one.
You can't go from that to actual percentages in base pairs.
Yeah, he's then trying to infer size.
With
Okay.
about 10%.
Well, right, but he says about 10%. If the real number is, I don't know, 1%, that's not about 10%.
Right.
Well, what would you estimate the number would be from those
in a real
pictures?
paper, when people say about, they don't say about. They give a number, and that's a mean associated with some standard deviation. Or if it's some Bayesian sense, it's a number associated with some.
Oh, really? So you think in all published papers no one's ever given an estimate of a number?
Yeah,
That's bullcrap, and you know it.
We give estimates of numbers. We give estimates. Ben, if you let me finish. We give estimates of numbers all the time, but an estimate of the number isn't about and then the number. It's the number.
Yes,
and
it's an estimate.
Some statistical measure of the spread of that estimate.
So basically you're saying that because he eyeballed it and looked at relative size, that's not legitimate because he didn't do a sequence?
It's very sloppy.
No, yeah, basically what I'm saying is that he does not have an accurate measure of the size of the chromosomes.
Does anyone?
Yeah, now we
Yes.
do. Yes.
And I don't think
We did
that
in
Tomkins would
1982.
believe that you did.
Well, he's
Because it's been matched arbitrarily
You're gonna
through the
echo
human
there.
genome.
somebody's got real bad echo.
Yeah. Herman, you're getting a little
Am I
echo.
echoing?
Yes.
How do I do? Hold on. I got it.
Okay.
I'm new.
No worries.
All right. Well, anyway, this is just a figure, and it's not intended to show an absolute size, and he never claimed that it does. And all you're really arguing is that, well, this sequence is bigger than it should be, so you're also claiming to know that this is in the exact size that it should be. And I already explained to you why Tomkins would say he doesn't believe that. He doesn't believe it because the chimp genome was matched onto the human genome, and he also showed me a paper from, let's see, 2011. Let's see if I can pull that up, of somebody who did a study searching for human-specific sequences, the LUY sequence, in other animal genomes, and found a whole bunch of them. So why is there human DNA in these other animal genomes?
Well, let me ask Chris.
Because they're using the human genome to map these other genomes.
Let me ask CRISPR real quick. CRISPR, if you did that diagram that I don't remember the software they used for that, but if you took that diagram and you actually did it with the actual exact measurements using the base pairs, base by base, would the drawing that Tomkins had be more representative of what we would find, or the one Ruhif did when he actually did it by using base by base?
Well, I mean, that is the way that Ruhif did his. So,
and he has a methodology on there too, right?
Yeah, and not only
I can't find it. I don't. I know I have it somewhere. I don't. For some reason.
Not only that, but Ruhif included the actual code that drew his figure. So, which is always a nice thing to have.
Okay, so the contention is basically that the drawing is a little bit exaggerated.
Well, no, there's something more than that. There's a more important point here, which is that he uses this drawing to come up with his number of 24 million bases of the size discrepancy. Okay, so he then talks about 24 million missing base pair, right?
Which hasn't been established.
Right, which he got from drawing this figure that he drew using completely inappropriate methods, right? So,
you don't know what methods they were. You're asking me what they are.
I'm sorry.
I'm sorry. I don't know it well enough to. I have some notes here where he lays out some numbers. Would you like me to read off the numbers? I don't know.
No, I'd like to make this quick point. The reason that I don't know the methods he used is because he didn't include a methods section.
Because it's not super important to his paper. His paper is primarily to report a binding site for a transcription factor.
Well, this is one of the things why I didn't want to call it a paper, because to be a published paper, they would have to have a methodology section, right? I mean, there has to be a method section so people know how to...
If you're primarily using computer programs to search public databases for sequence information, then you cannot publish the computer program. It's impossible.
Yes, you can.
You can put it independently on a website or something like that.
You include it in your supplemental data.
Well, from what when I talked to him, he told me that he had put all the programs that he's using on a website, and if he's here, then next time maybe he can do more of that. But he did alter the BLAST program a little bit and mess around with the parameters, and the reason he's doing that is to try and get rid of all these ends that are in there, for instance, sequences of the chimp genome that aren't sequenced, trying to align sequences directly without the evolutionary assumptions going into it, and using the human genome as a framework.
Did you happen to ask him, or can you ask him, why he didn't include a methods section out of curiosity?
I can ask him that. I've read lots of papers that don't have methods sections before. You know, you guys have these standards that you claim to adhere to, but the fact is I've read papers that don't have method sections. I've read papers that have estimates of numbers that are not important to the central argument, and all of that. The only reason you're doing this is because he's a creationist. Let's continue with Ruhif's
I want to say that's not really the case. I mean, everybody here actually looks at things very, very objectively, because this is what they do. I mean, so it really isn't because he's a young Earth creationist. When we've discussed Tomkins' work, we've done so with similar groups all from the scientific point of view. The fact is, younger the creationist actually doesn't even get brought up when we're talking about the actual data he's used for his claims.
So I don't know if this is going to mean anything to you. I don't make sense of it. Here's the notes that I wrote down when I talked to him about it. For chromosome 2A, the total length is 113,622, or 113,622,000, or 113 million, sorry. 113,622,374 bases, of which 7.2% are ends. That means they didn't sequence it. So the regular genome is ACTG, and an N means they don't know what it is. So 7.2% of chromosome 2A are ends that they don't know the sequence for. That means 8 million bases, corresponding to 5,575 gaps. Okay, with the largest, let's see, the largest gap of ends was about 3 million. 3 million ends in a row. For chromosome 2B, the total length is about 247 million bases, of which about 48.6% are ends. Almost half are unsequenced. And the chromosome 2B is the one we're looking at here. That's the one that has the cryptic centromere on it that is farther away from the fusion site than it is on the human chromosome.
Can I go ahead and jump in now?
So, sure.
Okay, so you understand that chromosome 2B had a large blank spot artificially included
in order to map it to the human genome. Right?
So the actual...
Which is what I said before, and you acted like that didn't matter.
No, it was done for computer reasons. Okay, if you're talking
in order
about
to get the computer to align the sequences correctly. Right?
The reason is that you're trying to start the very first base pair of chromosome 2B. You want it to start far down, okay? The actual length of chromosome 2B is 133 megabase. It's not 247 or whatever you said that number was. I'm looking at the data right now.
Okay. Were you going to continue?
No, I'm just saying that when he says that, if he's talking about it being 48% ends, he either is lying or doesn't understand how the system was set up, which either way is not very good.
Well, I don't. Again, I'm not a computer scientist, so I'm not going to be able to address the computer.
If I might jump in to give a practical example, if you student in your course, maybe you think plagiarized a term paper off another paper, and it's a 10-page term paper, and they left out a page of that term paper, and you line it up with the paper you think they plagiarized from, and every word lines up exactly, except for this block that they left out, then I think it's a fair case to say that they plagiarized the paper and just left out a big chunk. The gap there is an alignment gap as a methodological thing. It's not based on any necessary evolution. We're not like putting that gap in there just so we can maintain evolution. There just aren't the same number of bases in the two chromosomes.
That's an alignment issue. It's just to get it to align properly.
That's the whole point, is there aren't the same number of bases, and so it doesn't align properly unless you add a bunch of fluff and extra stuff in.
No, hold on. I got to jump back in here. So if you add the actual length of chromosome 2B to the actual length of chromosome 2A, and then you compare it to human chromosome 2, the two numbers are very, very close to one another.
And you're taking the data from the published Chimp genome.
Yeah, because it's contiguous.
So you're saying there's no ends in it?
Do you know why most of those ends are there?
I'm sure you would like to tell me.
It's very difficult to sequence using most technologies that are available, heavily repeated sections. Okay, so you know the sequence of this repeated region, but it's hard to say how many times it happens. Are there 50 50 copies of this repeat, 100
Right.
copies of this repeat?
Because
and so,
you can't use markers because it's a repeat. I get that. You have to use them outside the repeated sequence.
Right. So the point is, they know the sequence that's in that gap. They just don't know how many times it repeats itself.
Okay.
So they're not actually missing anything. They just don't know how many times this thing repeats itself.
So in other words, you don't know. And so what Tomkins did is he went to an older paper that used an older method and tried to extrapolate from an older wet chemistry method what the approximate size would be.
I'm sorry, what the approximate size of?
The approximate loss of chimpanzee chromosome that occurred around the fusion site. The amount of DNA that would have had to have been lost in order for the sequence to be the right length.
Well, here's the problem though. If you add up the sequenced length, right? So this is, get rid of the ends, the unknowns from the sequence length of both genomes, right? And you add 2A to 2B, and this is just what we've got sequenced, okay? And you compare that to just what we've got sequenced on human. They're very close.
What matters is the sequence we're looking at and what's been lost from the sequence that we're looking at, not the total length. So we're looking at a sequence basically between the fusion site and the cryptic centromere.
Right, and I have to point out here that he actually references a paper that gives the exact numbers on the size of those missing areas, and he then ignores the fact that they give those numbers and makes up something else.
Can you post that paper? I'll look at the paper.
While you're posting that,
Sure.
I'd like to welcome Fiona real quick. She had something to address real quick when it came to the methods section, if that's okay. If we can just rejoin that real quick. Hi, Fiona.
Hello, can you hear me?
Yes, we can hear.
Yeah, hello. No, there's something I wanted to address, mainly for the benefit of the audience. Is that something was said earlier on that the method section of a paper isn't important?
I didn't say that. I said I've read papers that don't have method sections.
Yeah, no, yeah, I think you actually did say it wasn't important, but I mean, it doesn't matter. What I'm trying to point
that's
out
not what I
is
said.
that, hold on, I think I've got it somewhere. But
Look,
here's the point. Here's the point about the method section. The method section tells you exactly how the experiments were done, exactly how they were done, so they can be replicated.
He's not doing a wet chemistry experiment. He's just running.
May I? Now, one of the reasons that the method section is very, very important is because the validity of the conclusions that you draw are dependent on you having actually done what you think you've done. And that's all down to the experimental design. Now, if you've designed your experiment incorrectly, then you may end up answering a question that you weren't asking, but applying that answer to the question you intended to actually ask in the first place. That is why a method section is very, very important, because when someone else reads through that method section, they see exactly what you did in your experiment. They can actually follow what is going on, what is actually happening at the bench when you did that work. And for that reason, the method section, it's not just methods, it's usually materials and methods.
He wasn't doing anything on a bench. He was running a computer program.
Excuse, but the point is this: whether it's at the bench or at the keyboard, you have to include your methods so that it can be replicated by other people, and they can then either confirm or refute your conclusions. that actually happens after the peer review process.
Well, in this case, it happened when Ruheef peer reviewed it and found a bug. So it can happen outside the normal peer review process just as well as it can within it.
I didn't say within. What I said was that normally that replication business of the actual, the actual experimental work, that replication usually takes place.
I think that Dr. Tomkins. Sorry.
That process usually takes place after publication. After publication in a peer-reviewed journal. The problem with what Dr. Tomkins has produced is that we cannot replicate what he did because he didn't tell us what he did.
Correct me if I'm wrong, Fiona, wasn't there some confusion a while back of what build he used too?
Yes.
Didn't Sam have to kind of delve into it and kind of do a lot of legwork to figure out what builds and things he was using?
Yes, and so I just wanted to
2015 paper. I think you'd be happier with him explaining what he did.
Right. All I'm doing is I'm pointing out.
And I'll post that here in a second.
So all I'm doing is pointing out that the method section is one of the most important parts of a paper because it tells you if the conclusions that the authors come to.
And all that I said, Fiona, I'm not denying that. All that I said is that I have read papers like this before that are using computer programs that you simply cannot publish the program in the paper.
No, I'm not suggesting you publish the program.
It's just a ream of meaningless code.
No, right. I'm not suggesting you publish the program. What you do is
you do publish the program.
You publish it on a website and you post it so other people can use it.
There's repositories for the source code.
He was using BLAST-N, which is a publicly available program, and that's how Ruheef critiqued his paper.
But BLAST was a published thing at some point too. You just cite it. It's something that's used. And that's not the point. It's how he used BLAST and how he used all these techniques.
The methods of how he used it, what he did.
Yes.
Look, I'm gonna post his paper from 2015, and that has a lot more detail about the stuff that he did with the computer program, which I honestly know nothing about.
Can we move on
to the paper? May I make this clear, please?
Sure.
I am not suggesting that Tomkins should have published the code to the entire BLAST analysis. That is not what I'm saying. But what I'm saying is you publish your methodology, how you've done your comparisons, etc.
And what parameters you use.
So, exactly. That's the methods I'm talking about.
If that's what you want.
I'm not talking about the entire coding for BLAST. That is not what I'm talking about. Now, if somebody comes up with a brand new method, a brand new program for doing this sort of thing, then of course they will publish their code. And then other people can look at it. But at least when you're doing something like these comparisons, you have to see how you did your comparisons so that people know what you're actually comparing, the parameters that you put onto your comparisons, all of that, so that people can then replicate your data.
Can you check out the link that I posted? That's his more recent paper on this topic from 2015, and I read that one as well, and it has a great deal of detail about the parameters of the searching that he did, the different method, the different versions of BLAST that he used, and everything like that.
Yeah, and that's published about the anomaly, and that his previous result.
He reran the results several times as part of this study.
Exactly. But you see, that sort of error from 70% going up to 88%. That's
it's an error in the BLAST program, not an error that he made.
I know that. Please, if you would, please stop interrupting me to argue against a point I'm not even making. That's almost a 20% difference. That should have been picked up. That there's something far wrong here.
Yeah, there's something wrong with the program.
Right, but shouldn't Tomkins have gone? This is way off from what the current thing is.
Steve. that he should have picked that up. Rue have picked up on this problem. Now, and it would probably be picked up by an awful lot more people if the full methodology had been published, so exactly what had been done.
What is the current value at? Do you know, Fiona? Is it like 98.5 or something like that? 99%?
Off the top of my head, I can
somewhere in
tell
that
you,
ballpark. Are you saying that when he got 70, he said Warning Bell should have gone off and said, you know what, something's not right here?
Yes, I think so, because what you have to do is a good control for it is just run a human sequence against a human sequence from a different individual and see what figure you get there. And if you get 85%, you think, oh, that's a bit odd. If you try running the sequence obtained from one individual with the sequence obtained from the same individual but in a different actual wet experiment, and it doesn't come out at 100%.
These aren't actual experiments.
Sorry?
These aren't actual experiments. They're just computer programs running sequence analysis.
But what I'm talking about is how you obtained the sequence in the first place. And how you obtain DNA sequence is by doing wet experiments.
I would agree with that, and that's what
you
the
agree
and that is okay, and that is what I said.
So Ben, you would agree that a good control would have been to do an analysis to like 2 humans and see, you know, if you're getting 85 between 2 members of the same species, 2 humans, that would probably indicate a problem, right?
And also, that's why I also said that if you take sequencing data from one individual and the sequencing data from the same individual, but obtained in a separate wet experiment, and compare those two.
In this...
Well, let's move on a little bit from there. Do you want to continue on the paper?
In the 2015, I just want to say, in the 2015 paper, he did exactly that. He ran the program several times, different versions of it, and compared. I believe is what he was doing was comparing human to human, and got 100% to make sure that it was working correctly. So he is taking these things into account, and he is recognizing the issues and dealing with them. His main issue is what I tried to explain at the beginning, is that he does not trust the published Chimp genome because they didn't do a separate genome linkage map to map the shotgun sequencing technique, which is what they did when they sequenced the human genome. They just used the genome as the Chimp linkage map. Okay? Now, I think that's a significant problem that is in a public database that is far more consequential than any mistakes Dr. Tomkins may have made. Okay, so...
Hey, just a quick question, though. You understand that the current sequencing methods, you know, you're talking about 50 to 60x coverage, right? Which means you're going to have the same sequence with a slight overlap several dozen times, right? So you're not just trying to align it to markers, but you're actually getting these overlaps that kind of slowly walk themselves over and over and over and over to form a continuous sequence, and you're getting it with 50 times coverage.
Mm-hmm. And if they had finished that, there wouldn't be any ends in the published data.
Well, no, there still would be, because these methods don't do very well on heavily repeated segments.
Mm-hmm. Which a telomere is a repeated sequence.
Yes, it is. I don't know what your point is, but
well, that's the sequence of the fusion site and surrounding it, supposedly.
Well, the fusion.
There's a lot of repeats.
Yeah, it's quite a few repeats, but when I say heavily repeated, I mean, let's say a section that's maybe a couple hundred base pair long that repeats itself several dozen times.
So not a telomere repeat.
Yeah.
Okay.
Although technically a telomere repeat would do that, but you have repeats within repeats, but that's just not really the point.
Right.
The repeating unit is a different size.
Yes.
A lot bigger size.
This is what I would say, in my amateur understanding of the issue, is how Dr. Tomkins would respond to this sort of thing. Is that ultimately he's running these programs on the published data and just in order to see what he gets with if he changes some of the assumptions around. But ultimately, I don't believe he trusts the published chimp genome. And he gave an example of a good study that he liked that did a good job, and that was a study in Nature that compared just the Y chromosomes. And he thinks that they actually underestimated the similarity, I think, on that one. Let me see if I can pull up that reference. But it was an example of, you know, not using the human genome as a framework to align everything, and then, you know, searching it and voila, oh, looky there, it looks just like the human gene.
All right, let's move on a little bit.
Question here. How do you compare two sequences, like a human and chimp, without reference to the human?
We're talking about how they mapped the chimp genome in the first place. It's a method of how they put the chimp genome together in the first place.
Right, and also, what has Dr. Tomkins done about correcting these errors in the chimp genome?
Well, I'm sure he would love to do something about it if he was able to access a couple, you know, a few $1 million grants from the NIH, who are not going to discriminate against him because he's a creationist. And he worked at Clemson. He was at Clemson for I was like almost 10 years as a professor. And what did I look up here? I actually don't know much about him, but you know, he was a full faculty member at Clemson, and I imagine that he left Clemson when he came out of the closet.
So let me ask you this then. So if you do a sequencing on a region with 50x coverage, right? And you have a continuous segment of the chimpanzee chromosome that's, say, I don't know, 10 megabase long or something like that. It's continuous, no gaps, right? And it's overlapped 50 times. And you compare that stretch that you've not used the human genome at all to do any alignment. It's just mashing it to itself because of these overlaps. And you compare it to the human chromosome, and you see that it aligns very nicely to a equivalently sized stretch of the human genome. Is it fair to say that those two correspond?
In terms of homologous genes, is that what they did? I mean, I don't.
Yeah, it's pretty much what they did.
All right. Well, I think I would have to defer to his judgment on that. But
all right, let's continue on the actual paper, because I still want to get to the centromere, and I definitely want to get it to the DDX11L2. So move on to the paper, to the second part. I believe he starts talking about the alpha sequencing, the alpha sequences in the cryptic centromere. Ben?
He referenced a paper by Karen Miga in 2016 when he talked to me. I can post that if you want. I don't think there was much in there that he was using.
I should point out real quick that you know, Sam has given us the paper. I put it in group chat, so it's Fiona from Nature.com, where the genome assembled was actually de novo for the chimp genome, according to this paper, which was published. When was this? I had it up, but then that one's from September 2005.
I'm trying to find that one where they compared the Y chromosomes. Here's the one from Nature where they compared the Y chromosomes. That one's actually free without a download. It's on a HTML.
Well, the Y is going to have a lot of differences, right? Doesn't Y have a lot of changes that go along with the Y chromosome?
Right,
it's not a very large chromosome.
And he told me that it's unlikely that that's the amount of difference that's there for the rest of the genome. But hello?
Yeah, go ahead. I just got some background noise.
Oh, okay. He referenced this paper as a method which he thought was a better method. So if you want to know like what kind of study he would do, I imagine it would look more like that one that I just posted there. Okay, so are we gonna move on to
Yeah, go ahead. Moving
what
on.
now?
I just got I got some background noise here. I've got to deal with. So move on the paper because he talks about the he wants to compare it to a sat DNA fusion, right? Can you talk about what he means by that and why he thinks it's a satellite DNA fusion rather than telomere to telomere fusion?
Oh, okay. Well, satellite DNA is found around the centromere, as I understand it, and all observed chromosomal fusions in viable organisms have occurred around the centromere, and it contain a lot of satellite DNA in and around the fusion site.
Except for pigs, zebras, antelopes, fish, rats, and a couple dozen others. But
okay, well, and do you have something I could look at there that I could show him that is for chromosome fusions from other animals? And you understand he's talking about stuff that's been observed, not hypothesized from a sequence.
I actually have a couple links on those actually. I'm going to give you the ones that Sam had used for some of the work that he's done on this. These are actually his links that he's provided. I think that's what you're asking for. One sec. Okay, I put him in the. Oops. Yeah, that's right. If Sam doesn't mind me reading this out, I've asked him before if I can use this. He had written something to Dr. Tomkins, and he says, in your introduction, you claim that a telomere-telomere fusion has yet to be observed in humans. However, several instances have been observed, the two papers that I cited there.
Oh, that's a 1988 paper. That's too old. Can't look at that anymore.
He says, well, hang on. He says both of these references show humans that have undergone a Robinsonian translocation, leading to a telomere-to-telomere fusion. Moreover, they predate your article and should be included in some sort of Corrium or errata section as they dispute a direct claim. So it is older, meaning that he's saying that it should have been included because these are something that indicate that there have been telomere-to-telomere fusions.
Okay. Well, I can maybe present that to him and see what he says about that. Okay. It's possible he hasn't seen those papers. I don't know. So he says that generally, so a telomere-telomere fusion, let's get back to that.
Sure.
This 800 base sequence, it is inside a functional gene that codes for a long non-coding RNA that is involved in the RNA helicase gene network, regulatory network, I believe, is the issue. So the idea here is, how is it that the fusion site, which is 800 bases long, was preserved, and all these other sections that are around it were deleted out? And also, how is it that you get two coding exons that are around this fusion site? So one coding exon comes from the chimp genome, and the rest of them are in the human genome, or rather, one is in one chromosome and one is in the other chromosome, and that doesn't make a lot of sense, considering that, again, part of the reason he's assuming a large difference between the ends is that telomere regions are long, and subtelomere regions in the humans are 5 to 15,000 bases long. And here you have a RNA coding, not protein coding, but RNA coding gene that, it has a very short intron sequence that includes the fusion site between exons.
Chris, do you want to address that?
Yes, I do.
Figured you did.
So just a quick point here. You would agree, of course, that for DDX 11L2 to support your case and Tomkins' case against a fusion, it would be necessary that all the data you present for DDX11L2 corresponds to the transcript that actually crosses the fusion site, right?
Well, yeah, that's a binding site that is in and around the fusion site.
Because there are multiple transcripts of DDX11L2, right? And only one of them crosses the fusion site, right? And so the ones that don't cross the fusion site are not relevant to your argument because they don't cross the fusion site.
Yeah,
right?
so there's two that you're talking about, I'm assuming. There's 3 exons in the gene, and one is the longer one that's farther downstream on the minus strand. And that's the longer one. And there's 2 short ones upstream. The first upstream one is on the side of the longer exon, and the second one. And the first one is opposite of the fusion site from the rest of the gene. And that's the variance.
Exons. I'm talking about transcripts, not exons.
Yeah, I think what he's asking.
Transcripts have the exons spliced together.
But I think what he's asking, if you're going to have evidence that is transcribed across the fusion site, then you need the evidence has to be from that specific transcription, not from the canonical one or another one.
That's right. That's right.
So you're saying that because they're not in the transcript, that it's not part of the gene, even if it's a promoter region?
No, I think he's saying that the transcript, if you're going to show transcript evidence, it has to be that transcript from that particular alternate transcript.
Yeah, basically, what I'm saying is that if you have evidence that the short transcript is doing something, right? Short transcript doesn't cross the fusion site.
Right.
So,
none of the transcripts would, because the fusion site is in the intron. It's being spliced out because all it's doing is a promoter region.
There are transcripts that include the fusion site.
No, that's not right either. But the thing is that there are multiple introns, right? And so the way it works is that you have an exon, and then an intron, and then an exon, and then the first half of an intron, and then the fusion site, and then the second half of the intron, and then another exon, right?
What I saw,
I may have had that backwards.
What I understood was that because this is on the minus strand, so it may not be going the same direction.
I may have said that backwards, but it may be backwards, but you would agree that that's an approximate diagram of it, right?
Let me just say it again to make sure we have it correct in the collective mind here. The first exon is, let's say it's on the right side.
Let me just pull it up on UCSC. Okay, that'll be faster.
Yeah, then there's the fusion site, which Tomkins is saying is part of the binding site for the transcription factor. Then you have, that's an intron that's going to be spliced out of the transcript. Then you have the second exon, which is the other short one, and then an intron, and then the longer exon. So there's only 3 exons and 2 introns, from my understanding.
Okay, you're confusing the word exon for the word transcript.
No, because exons are the only things that get into the transcript, right?
Yes,
introns are spliced out.
No. The thing is, one transcript might have exons 1, 2, and 3.
I understand that.
Let me finish. And another transcript may only have exons 1 and 2. Okay?
You're assuming that I don't know these things. There's
Because
2 variants.
Like you don't.
There's 2 variants. Okay? One of the transcript.
Short and long.
One of them has the 2 exons on the left. I'm just using left arbitrarily here. Left of the fusion site. The other one is the longer one that has the 2 on the left and the 1 on the right. The transcription factor, assuming probably for the longer one, is on the fusion site itself. So it's actually there. Okay.
Right. So you have the short transcript that does not cross the fusion site, and the long transcript that does cross the fusion site.
Basically, yes.
Okay, great. So you would agree that the short transcript, which does not cross the fusion site, does not support your argument because it doesn't cross the fusion site.
Well,
you
okay.
need a transcript that crosses the fusion site.
You're saying the longer variant doesn't exist, or you just?
I'm getting there. I haven't said it yet, but I'm asking you if you would agree that if the long transcript didn't exist, you wouldn't have an argument with DDX11L2, right?
No, I'm saying that it wouldn't be in the transcript. It would still be a transcription factor binding site.
Does somebody want to present this? I'll present them if they have the
One
Genome
second.
browser.
But
So
I
we
mean,
see if we can see
if
what's
you want to
going
go, I can go down that road. Just go ahead. I'd say, yeah, sure. Now, what's your point?
Well, my point is that all of the data for DDX11L2 is for the short transcript. There is no data for the long transcript.
In what?
So if there's no data for the long transcript, you have no evidence supporting your claim.
In his paper or in code?
In a database anywhere.
Or anywhere.
Yeah, here. Let me link this to you. This is the actual. Nope, that's the wrong one. Hang on. I think was it Ensemble that. Let me see if I can find that one. While you were finding the thing where you can show people for the genome
I just linked
database.
to the Ensemble site for that gene.
Did you?
Yeah.
Let's see here. Yeah, that's the right one. So I'll put that in the live feed because if you click on that, if you go to supporting evidence, let's see here, transcript table. I don't think there's any actual transcript for that particular long transcript, is there? Any transcription? Let me post this in the outside feed. Why don't you look at that, Ben, real quick on the Ensemble?
No, the transcription for the long transcript is essentially null.
So he's giving NCBI. This is not UCSC.
Yeah.
Yeah, this is what was posted on Ruhi's blog, right?
Yeah, the no transcript supporting evidence for this transcript.
That's in encode.
That's in Ensemble.
Ensemble. And this is basically
actually link the whole
transcription or transcription levels or expression levels. Is that what this is?
Mm-hmm.
Hey,
Okay.
Steve, if you want to go ahead and present
So-
me.
There you go. Let me present you real quick.
So this gets to. Go ahead.
This is a pseudogene, right?
Yes.
It's not. I mean, what's its function? I mean, they're claiming to be some function, but it's, by everything I can see, it's expressed as a pseudogene.
Pseudogenes are being found to have lots of function, and part of it is based
What's this
on
one?
it's.
What is this gene's function?
Hold on, we're not even there yet.
We don't.
Can we talk about what I've got on the screen?
Yeah, that's going to be jumping ahead a little bit about the functionality. So, Christopher, you have the UCSC browser on your
Yeah.
screen.
Okay.
And
So
I'll put
you
that
see
in here.
the one on top here. This is the short transcript. Okay, and you can see that it. Firstly, you might notice that the exon sizes are different between the two transcripts, right?
So the exons are the blue ones.
The blue bars, and then the dotted lines are the introns. So you see that there for the short transcript there are 3 exons, right? And then the fusion site is over here. Okay.
Right.
And then for the long transcript, there's a little short exon here, and then a long intron, and then another exon, and then right. So now we're on the other side of the fusion site, right? And here are the other 2 exons. So the point is that for the short transcript, and this is the one that actually has transcriptional evidence. It doesn't cross the fusion site.
Meaning what?
Meaning
Expression
that,
levels?
yeah, it's been detected at some fairly low level for the most part. But so the long transcript, which is the one that crosses the fusion site, hasn't been detected. Okay, it's a hypothetical transcript.
Okay.
Now you would agree that if something isn't actually expressed, it doesn't have a function.
Okay. So when you do these big databases, what tissue cell type are you using?
Well, I can pull up.
I have the GTEx.
GTEx here.
I have it right here. Actually.
GTEx will tell you. GTEx gives you all the results.
Okay,
And
so
the only detection at very low background levels, basically, was testes. Everything else was basically
Actually, Fiona, it's worse than that. You're just gonna have to give me a second to get it loaded.
Yep.
All right, I put the.
Is this again from Ruhif's blog? So the part where
No,
he
this
shows
is
a little
from the
red
databases.
dot.
No,
this
actually,
is
I
from
think
the
I did
databases
this.
themselves.
All right.
Yeah, I put the link in there so you can look at them too. This is actually from the databases, and this, correct me if I'm wrong, but isn't these are the same databases that Tomkins uses in his paper, correct?
He's using the US UCSC and NCBI.
Does he mention the GTEC? I don't remember.
The what?
The GTECs. I don't remember where he got his expression levels from. But it doesn't matter.
It looks almost exactly like. Did you just take that figure from his paper?
What figure?
The figure he just showed me.
What we're looking at now?
No, both. What he has just showed you came from is the ensemble database. It's not from Ruhif's blog.
Yeah, that's from the database.
Okay.
And now the data he's now showing is from the GTEx database. Not from anybody's blog.
Okay.
Tompkins does include the USC. He does include the
everybody-
UCSC browser in his paper. Exactly what you were showing. That's what he has.
Can everybody see what I've got up here?
Sure.
Okay, so I've got this by isoform, right?
Yes.
So these are the different transcripts. You see the red line? That's the short transcript. See this green line? That's at 0 all the way across? That's the long transcript.
Okay, so what tissue is it in? What cell line was
all
this
of these.
done?
0 across the board.
Which ones are those? Are those
Whole blood. And
adipose, adrenal,
Yeah.
artery.
roll over.
okay? So what his expression data, it looks like maybe from biogps.org. And they listed significant levels of expression data in 16 out of 31 major tissue types.
For the short transcript.
Right, for the short. The long one has 0. The short one, it actually has some measurement in adipose, visceral, I can't read these. Adipose, visceral, adrenal gland, artery coronary, artery tibial, bladder, etc. So there is like low. It's like what are some of these 1.5 RPKM, 0.7 RPKM? I mean, they're really, really low.
They are basically background levels. If you look at the error bars, they are background. They are so low that you cannot say that is expression. So if the gene isn't expressed, if the transcript isn't detected, if it is not found, then you are not talking about a real life transcript. You're talking about a hypothetical transcript.
It's hypothetical. It might be hypothetical, but it's based on a transcription factor binding site that's on the.
It may be
that's
that's
on
great,
the fusion
but
site.
Even if there is a transcription factor binding site, if it isn't transcribed into a transcript, then you cannot talk about said non-existent transcript.
Okay, well, I can show you another paper that he showed me, and that is how long non-coding RNA often do not have significant expression levels.
They have to have
And that's-
some, though, right?
They
Yeah.
do, so for instance, this one is a paper about granulocytes, and I just want you to look at the purple bar graph that is in figure 2. So if you, I don't know, I posted the link if you guys can see it. There's a bar graph there with that shows granulocyte specificity, where 4% of mRNAs that were expressed in the granulocyte were in the public databases, or the public annotations, which on the graph it says PA for public annotations. That's the public data that you guys are looking at right now. And 11% of long non-coding RNAs that were expressed in the granulocyte are in the PA. But there's a massive percentage of long non-coding RNAs that are expressed in the granulocyte that are
But we
not.
point out these aren't expressed.
But
We can't
also-
find the message. Nobody's detected the message. The message is
Well,
not
and
there.
in order to in order to detect it, you would have to do a similar experiment to what these guys
But
did.
can I just point out a very important thing here, which is that the fact that something is transcribed doesn't actually demonstrate that it has a actual function, okay? It
Exactly.
needs to be transcribed to have a function, right? So if it's not transcribed, then it
you don't
doesn't
need
have
to
a function.
point that out to me.
So, but the thing is, you just showed that, well, there's a lot of long non-coding RNAs that aren't in the databases that are transcribed at very, very low levels. So,
Yeah.
do any of them have a function?
It
Who
means
knows?
if they're expressed in tissue-specific places, what it means from a genetics perspective is that something about that tissue, the organism, it's accessing that portion of the genome for a very specific reason. Okay, that's why tissues are different.
But
So, for
does it
instance,
mean that that given transcript has a function?
no, you have to elucidate it all individually. That's why sequencing a genome it doesn't really tell you a whole lot. You have to do the wet chemistry on all of that.
So we're
But-
agreed that
but-
expression does not demonstrate function.
But sequence often does, and sequence of a promoter, things like a promoter, is genetic evidence that there's a transcript there.
No, that's not true.
Yeah, that's why it's a hypothetical transcript
Yeah,
for that very reason.
that's the point. You can have a gene that has been completely disabled because a huge chunk of it is missing, but the promoter and the beginning of the gene is still there. But the point of this one is we haven't found a transcript. It's not been detected. It's not there. So nobody has found the transcript. People have looked for the transcript. They haven't found the transcript. So you can't really, as I said, you can't really say anything about this particular transcript that so far doesn't exist.
I doubt that people have looked for this particular transcript, but you'd have to do so with a specifically designed methodology just for that single one, which is just like the paper that I just showed you.
Yeah, you would find it
because you have to know what tissue it's in, where it's, and all of that. It's.
Yeah, and that is why CRISPR has just shown you a table showing the results from all the different tissues, and it wasn't there.
Well,
CRISPR, could you pull that up again?
I saw it the first time.
Right, so the point is the methodology for looking for it is to use a probe specific for it. That's easily designed.
These are it's not easy. These are from public databases that are thrown across the entire genome all at once, and all the data is just thrown up on the web there.
Where do you think the data comes from? It comes from people actually having looked for it. They've actually looked for it and they've not detected it.
So they've done like a.
Could you put that thing up again, CRISPR, please? That table, the bar graph. And the point is that, yes, it is quite easy to detect a message, especially if you already know the sequence. It's very easy to detect a message.
Well, has someone looked at Tomkins' paper? Is that what?
Here we go. Here's the bar graph. You see the bar graph? That is what was detected in each tissue type. Detected.
By someone who's specifically looking for this transcript variant.
Yeah.
Someone actually looked at this hypothetical transcript that you don't take seriously because it's from a creationist?
The x-axis on the bottom, I'm assuming, and somebody correct me if I'm wrong, those are all RNA-seq data from all those different tissues. And they don't find that transcript in any of those RNA-seq databases that are created using RNA-seq for all those tissues.
And that is detecting.
Right. And these public databases, they're not specifically designed to detect specific things. And the whole point of this,
Those are databases, RNA databases.
because you have to go with co-expression levels and things like that. And the whole point of the paper that I posted in the conclusion.
I'm sorry, go with co-expression for what?
Seriously, can I just sort something out here? Ben, nobody's saying these databases are looking for it.
You just did say that.
No, I didn't. Databases don't look for it.
People scan the database.
Right.
Yeah.
And you scan.
Please.
I understand where the information from the database is coming
Sorry,
from.
I give up.
These are all RNA-seq data from all these different tissues. So you sequence all the RNA that's expressed in those tissues, and you put it in the database. When you do that,
so
you don't
you have to
find...
do the wet lab work.
Right.
I would agree with that. You have to do the lab work on that specific sequence.
And hold on. Actually, you're missing something very important. The paper that you gave us, Ryan, they used the same technique. They just read a lot more RNA. Okay.
Okay. What their conclusion was is that there is a very high variability of long non-coding RNAs in their expression levels, including significant functional RNAs that they showed in their paper that is functional for other reasons that are not detected in public databases like this, and they explained reasons why they might not be detected, such as tissue specificity, low expression levels. So, for instance, we know that there are functional. This might be a little outside the purview here, but we know that there are functional biological molecules that have biologically significant activity at expression levels that are so low that their binding is basically stochastic, meaning that it's basically random if they ever find a binding target or not, and yet they're still doing something. Okay, so that would be far lower than this type of level expression. And so if...
But wait, the claim was that it was highly expressed.
Right, and that's a big thing in his paper. Well, in the title, even it says.
Where does he claim that it was highly expressed?
Inside
a highly expressed
gene. I wish I could stop.
The whole gene is highly expressed,
but it's not.
No, it's not.
The name of the title of this thing is alleged human chromosome 2 fusion site encodes an active DNA binding domain.
And you guys are saying
and highly expressed gene.
You guys are arguing that one of the variants is not highly expressed, and he agrees with that.
No, he claimed it was.
No, none of the variants are.
He wouldn't agree with that because I gotta do this because the variant that isn't highly expressed is the variant he needs to make his argument.
The long transcript.
I understand that.
Right, so it's not highly expressed.
It's not untrue that it's within
even if the short transcript were highly expressed, which I'm not sure I agree with, but even if that were true, one, it wouldn't mean the short transcript has a function, but it sure as hell wouldn't mean that the long transcript is highly expressed or has a function. Okay, you can't just conflate these two things like it doesn't matter, which is what he did, which is why, well.
Sam, did you want to add to this real quick? I mean, because I think what it is now is basically. Do you agree, real quick, Ben, before Sam jumps in, that there is no evidence for a transcript on the hypothetical alternate long transcript? Do you agree? There's no transcription
agree with that.
Do you have a database is there a database we can look at that shows a transcription level for that?
I think the evidence is the promoter region.
Well,
why is it just because there's a promoter region?
The transcription factor binding.
We need to see the transcript.
I understand that.
Evidence that it is. We want evidence that it is transcribed, as you have just claimed. CRISPR's already shown us that there is no transcription there.
I mean, you can't say something has transcription just because it's a promoter, right?
Where is your evidence that the transcript exists?
Okay, I think that's a fair question.
Good.
Not
But-
the promoter site, the transcript.
I think this sort of illustrates a difference between how creationists and evolutionists approach biology and science in general. Okay, as a creationist sees something like this, and we see function and purpose, and we're more likely to believe that it is going to end up that it has a function if you examine it further. Whereas an evolutionist is the kind who, just as you just told me, is going to say, well, we don't know this. We
Well,
don't
Ben,
know
he is
that.
claiming,
And
though, it's
No,
expressed.
that
No,
is
that's
not
not
what
true.
anybody said.
It's
But
perfectly
Ben,
logical and reasonable that something would be
But Ben, he's claiming
transcribed.
that it is highly expressed. He's not saying that it's possibly highly
saying
expressed,
the
or we're waiting for more
gene-
information.
The
He's saying
gene
that it
is
is.
highly expressed.
Which
What can you define a gene for
Yeah,
me?
the gene coding for the longer transcript.
Not just
That's what he's
that.
claiming.
There,
He is claiming the gene for the longer
the
transcript.
definition of a gene.
Will you stop interrupting me when I'm talking, please? He's claiming that the gene, which encodes the longer transcript, is highly expressed. And CRISPR has already shown the bar graph that shows that it is not. Now,
The title.
we would like to see the evidence that it is highly expressed.
The evidence is that it's a transcription factor binding site. What he said
No,
was he
that's-
encodes an active DNA binding domain inside
That
a
doesn't tell us
complex.
it's a transcript.
Let's say for
You're
argument's sake,
correct about that, and I think that he went into a little more detail when I talked to him. But
let's say this gene.
So can I say
And-
something? Because I've been waiting, and there's
Yeah.
something
Go
germane.
on, I'll mute myself.
yeah, Sam, go
okay.
ahead. Hang on, Herman. We're
I
gonna.
don't need like 7 people telling me, okay, one person is fine. I'm sorry, I'm just being sarcastic. But Ben, you do realize like one of the big problems with transcription factors is that they bind very stochastically across the genome. And so like there's the issue of where there are a lot of non-specific factors. And if you want to say that something's functional or that something is going to confer activity, you need another analysis other than just saying there's a binding signature.
I would agree with that.
Because-
But it is evidence. So
it's not evidence, though.
I once
it's not evidence to an evolutionist who thinks that all of this is just random.
No. Like it's not evidence to a functional genomic scientist, right?
Let-
Like
let
this
me-
is what
let
I
me throw in
actually
one.
do for a living. So
Look-
I'm just telling you from the perspective of the literature. You're welcome to assert what you want to assert, but you need, other assays to say that a site is
a
functional.
quick point.
depending on the claim.
A quick point for context, and Sam, you can correct me if I'm presenting this in the wrong way, but I once put a completely random sequence, maybe 1,000 base pair or something like that, just completely random, through a binding site motif search, and it found several hundred perfect matches and many more close matches. And so this
How
is just a
long was it?
random
How long was the sequence you used?
1,000 base pair,
Okay, that's interesting.
right? And also,
Yeah, I mean, again, this was why I was hinting at it, and there, like, I can recommend a variety of approaches here, right? Like, you could do something like an EQTL analysis, or you could actually tie it to a regulatory network.
Well, one problem
Absent
with
of downstream analysis, I mean, all you have is a binding site, which is rendered as, it can be used in an aggregate analysis, but as a single data point, it's regarded as relatively useless.
one thing about creation scientists is they don't have the resources that other people do, so they're not going to be able to do that kind of a study. So the way that you would show this is would get this to be looked at by someone who has the resources to do a wet chemistry study like that. But okay, one thing I would like to, this is more general, do you guys, someone define a gene for me? I think from one of the big discussions that's going on in biology now is, does the term gene even have a meaning anymore? And part of the reason that that's a discussion is because of all the non-coding RNA that is being found to have function. And as part of gene regulatory networks that regulate expression levels in very complex ways. And so when one transcript of say an mRNA that goes to a protein gets out there, it's because a whole bunch of other RNA transcripts have been, transcribed and spliced it in a certain way, and all kinds of stuff. So my question is, and this is mainly, to anyone really, is there a working definition of a gene that sort of takes that into account? And that's an honest question.
I can actually answer this if people don't mind, because I actually wrote the last chapter of my master's thesis about this.
Awesome.
So you might have, and what it was about, if people are, and we can talk about this off air, or if I'm even here off air, but it was about cryptic translation of long non-coding RNAs, actually. But generally speaking, a gene is regarded as a heritable unit that is either transcribed or translated.
Okay, so would that count for multiple protein products that are regulated by the same regulatory network, for instance? That are linked.
What do you
maybe
mean by that? You just used kind of two different terms. So do you mean like splice isoforms?
Not just alternative splicing, but completely different coding genes that are coding.
Well, those would be separate genes.
But what if they're controlled by the same regulatory network and co-expressed?
They'd still be separate genes, because we're talking about different particulate molecules that are being transcribed that are the result of different loci.
Could I just throw in here?
So it's the loci.
You talk about different genes that are under the control of the same thing. Have you heard of the term operon?
Mm-hmm.
Right, that is multiple. Right, that multiple genes under the same controller.
Mm-hmm.
So their expression is all controlled by the one thing. So they're all expressed together.
Right. So if for instance, one protein coding region can be alternatively spliced into a bunch of different protein products, like 10 or more, and many more, what is it about one protein product that makes that a single unit?
They're not a single unit. The different splice isoforms are still the same gene.
Right. So
remember, you asked about gene, not about protein product. You asked about gene.
Yeah.
I'm asking how to define it.
Well, yeah. I mean, again, it's like it's one contained heritable, or like a set of loci that map to, products, right? So different isoforms are still contained within the same, even though there are different mRNA isoforms, it's still the same block on the DNA.
So physical location on the DNA.
Yeah, and again, I'm generally speaking, right? Like if we were to get down and dirty with it,
so would that include?
Aren't as concerned with a specific definition of gene. In
my
experience.
Well, right. You want to know what's happening. The words you use don't necessarily matter that much, but would that include promoter regions that aren't transcribed? Would it include?
They're regulatory genes.
Promoters are transcribed. I don't know what you're talking about.
Sometimes they aren't.
I'm literally it was funny when this hangout started. I was actually literally doing quality check analysis on sequencing data for promoters and enhancers. So I know for a fact, because otherwise I wouldn't have funding.
So you're saying all of them are transcribed?
Not all of them, only the active ones in that particular
I didn't make a general statement saying all of them aren't transcribed. I'm saying, are the ones that aren't transcribed part of the gene? What about histone markers? Because a lot of times
aren't part of the gene.
But you can't access it without finding the right histone mark and unfolding the DNA in the right place and finding the gene.
That's not part of the contiguous genome, right?
That's not part of the genome, though.
Okay. I'm just shooting shit right now. So
I wanted to get back to something.
Yeah, you are.
Can we let Herman? Herman, can you go ahead and say what you're going to say real quick? Herman's kind of new to the format here, so.
Yeah, I know. I'm trying to be cool.
We got a lot of newbies, so go ahead, by all means.
I'm a noob, but I wanted to get back to something that has to do with the actual chromosome 2 fusion site, and these genes are. These DDX11L genes are gene family that, except for the ones at this fusion site, they're always found sub-telomeric. So they're always found near telomeres. So here you have a gene.
Well, it's not near the current telomere.
It's near, right.
He said except.
Right, except two. All the rest are
the very existence of this gene is itself evidence that this is a fusion site, because the only other place you find these genes are near telomeres. So I don't understand why. I mean, let's say it's expressed. Let's say next to this thing, and it's functional. Well, nobody says it has to be, lose its function necessarily. The fact that it's there is a piece of evidence that supports the hypothesis that there's two chromosomes have fused in our evolutionary past.
And there was something like, how many other copies of it are there in the genome?
Well,
it's like
that's
a 10 or 11, is that correct?
The family is roughly the genome.
Was that how many
11.
approximately?
Here, I'll put the link on here to the paper.
But I think what Herman's saying is that
all
of them are subtelomeric except for this particular one that are right next to the fusion site.
Right.
Yeah, that would seem like evidence.
Exactly. So that's where I don't get Tomkins' argument using this at all. I mean, whether it's expressed or not expressed is, that's a valid discussion to have. But the point is, we're talking about a gene being expressed or not expressed that everywhere else is only found near telomeres. So that itself is a sign that this is a fusion site, an end-to-end fusion site.
So you think it's a valid line of scientific inquiry to look at whether this is expressed or not? That doesn't sound like what I was hearing.
Why wouldn't it be?
Why wouldn't
Yeah,
it
why
be?
wouldn't you?
Well, I thought you guys were coming after me on that and saying this is not how scientists do it. They look at the database, and if it's not there, they don't ever look for anything.
Well, he wrote a paper.
Hold on. If somebody wants to and can manage to get the funding to do that specific research, good on them, right?
But you're never. Creation science is
well, they do have a lot of funding. ICR is not poor.
Do you know the way that you get a grant to do that study?
Proposal.
You make sure that you write a grant proposal that removes all references to young Earth creationism. That's how you do it. That's part of how you.
Well, why would it need to have any reference to it anyways? I mean, if you're
because the reason he's doing this is to refute the human chromosome fusion argument. If that's the purpose of his study, he's not going to get funded for that.
I think
I don't really know why he needs. Well, can I ask a question? Like, I don't really know why he would need a huge grant to run something like a northern blot. Like, those are extremely cheap. Like, we have sophomores who run them,
but
at the university.
Actually, that's so. Any idiot can write a grant application that has nothing to do with young Earth creationism, right? And they still won't get funding on it because it'll still be a shitty grant application. Okay.
Right, and your contention is that Dr. Tomkins would write one of those instead of a good one.
That's not my contention at all. My contention is that if you want to get funding to do it, what you need to do is have evidence. You got to start that there might evidence that, hey, this thing might do something. Look at this evidence that we have that it might do something. We should take a closer look to see if it actually does it.
And primarily
funding, and then you do your research, and then you publish that research.
Right,
and you can show that it does something.
And it has to relate to cancer or some major problem that is going to get funded by people, or evolutionary theory.
I think you're kind of making an argument to motive here, right?
Well,
I mean,
here's the thing, right? Like,
science includes human motives.
Well, how much do you know about the funding process? Because, like, what you just said kind of seems to reveal that you don't know that much. Like, there's quite a bit of funds devoted to basic science. If he, like, I will say that if he tried to write a grant just based on this one gene, he probably wouldn't get it because it's just too small in scope. Like, why would the NIH or the NSF give him hundreds of thousands of dollars for something of that small scope? But if he was able to get, like, something where he was, like, hypothetically, is just shooting from the hip, like exploring, functional relationships throughout the genome that are unexpected or cryptic or something like that. Like, I could envision a case where if he wrote it well enough and he grounded it well enough in literature, he could actually get a grant going.
Yeah, and he would have to make sure that he doesn't tell anybody the reason why he's doing the study is to refute the chromosome hypothesis, which he basically have to lie about what his motives were.
No. You're wrong about this. I'll tell you why. The before I came into academia at my present position, I worked for a nonprofit. Actually, at those nonprofits, those people have development offices, and they can court donors to pay for whatever they want. In a lot of ways, to get sort of small amounts of money, and he doesn't need much money to do this. It's easier for him to get money than it is for me to get money. And I say that as someone who worked in a nonprofit and got support from private donors to do the work that I was doing.
How much money do you think ICR has? I have no idea. I'm just curious. You seem to think they have a whole bunch of money or something.
We're talking about what he wants to do. He might need $10,000, which is nothing. You can get donors to drop $10,000, especially someplace like ICR, in a heartbeat. So he could do whatever he wants. I mean, he could do whatever he wants.
He could do a complex microarray study of expression levels with $10,000. I don't think so.
Yeah, you buy a single chip. The chips for this microarray is about, what is it, $400?
Yeah, I'm going to turn on my camera.
That's about right, yeah.
Yeah, microarrays are ultra cheap. Like a lot of these things, I mean, it just kind of, or you know, from what I read of Tomkins' record, he actually was, you know, very good at prepping bacterial artificial chromosomes in plants. Like that's
and
like his back work is very good.
Way.
Yeah, like and I'm coming forward with this, but if he has, you know, from when he was a postdoc, if he still has some connections, I'm sure that, you know, he could get, you know, someone with an actual setup to either do RNA-seq or a microarray if it's really that dire straits.
Or he could be really nice. He could be really nice to someone and say that, you know, he'll help, that he'll work in their lab, right? And then just maybe, can I use this equipment just a little bit, right?
But you do acknowledge that if he was open about his goal, meaning to disprove.
No, I completely disagree.
From people who share his goals.
You share his goal to disprove that there was a fusion site in human chromosome 2.
Right. I mean, isn't that how science is supposed to work?
Wait, can I point something out?
So you should do the study.
May I say that if you put in a grant application, the aim of this project is to disprove such and such, then that will be knocked back, because you're showing you have already made up your mind on what you're going to find.
Well, yeah, that's the thing. So is if I was to propose this, I would propose it as this may have implications on such
exactly.
and such topics.
I'm sure he knows how to word it.
This in my hand.
What is that?
What? I'm not looking at it.
That is a DNA sequencer. It's a minion.
Oh, is that the new one that came out? That really badass one?
That's a minion.
It costs $1,000.
Yeah, we read an article on that. It was like amazing.
It costs $1,000. Inside goes a little flow cell. It's not in there now. It plugs into a desktop computer. You can sequence DNA by the millions of base pairs.
Amazing.
You can they're working on protocols now where you can directly sequence RNA from it.
What's it called again?
A minion. It's like spelled like minion.
Who makes
it's an ion. It's based on nanopore sequencing, where the molecules.
What's the error rate on that? It's not to derail.
The error rate is about you correct a lot of the error by coverage, so you can get a lot of coverage if you're doing like a transcriptome or something that's not a whole genome. Mm-hmm. But the error rate for the 2D reads has gone up to about 95.
How much was that minion read worth 30 years ago?
That's not bad.
The accuracy has gone up to
that's
like
not bad with scaffolding. That's not horrible.
And this can produce long reads, so you can do like sequence through repeats, because you can do like 20, 30, they're talking about 100,000 base pair read lengths on this.
Yeah.
Which, by the way, the great thing about the error rate there is that you don't really care when you're trying to go through a repeat. You don't really care about the specific sequence so much, because you're trying to figure out how many times the thing repeats in the first place.
Right.
So you're saying
seriously awesome tech.
The point is, he can get the money for this. He could have a line of donors coming up to ICR who share his goal, who could pay for one of these out of pocket change, and he could do it.
That's like Star Trek level technology. I mean, can you guys imagine 30 years ago if you would propose something like that? People would look at you like you're crazy.
They're going to build one now that plugs into a smartphone.
That's amazing.
Yes, I saw that. That looks fascinating. But you can actually do DNA sequencing in the field.
We're sequencing whole mitochondrial genomes on this right now.
Yeah, and that was one of his concerns that he voiced to me, is that the techniques that are being used are using the scaffolding from the human genome. And that is very difficult to do independently every
What
time
is
you
he
sequence a
doing?
genome.
He's complaining
What do you
about the
mean
method.
by that?
Because
what are you? Hold on, wait, hold on. What are you?
Can I ask, what does he mean by that?
That's what I'm trying to ask
Yeah.
too. So yeah,
Yeah, what does he mean by that?
you said the word scaffold. What do you mean by that?
Well, I mean it in the traditional sense, right? So when you
Can I-
get
Can I
get
take
a
crack at it?
Hang on, CRISPR. Hang on.
well,
on, Seb.
when you get a read from a sequencer, right? It's usually, okay, let's just say like 50 base pairs. That's a fairly average, on the smaller side of average read from like an Illumina sequencer.
200
and-
anymore.
Yeah, I'm talking about like small RNAs, but like if I think like the usual read range without the adapter is somewhere around, like if it's on the higher end, around 100 base pairs, but that's not important.
It's more than that.
And I can look at that. I actually have an email that has this, but
They're running
the
200s
point
now,
being
yeah.
is you assemble a scaffold, you have reads, and these reads overlap each other.
Right.
And what happens is,
Yeah.
is you can imagine, it's like if you were trying to reassemble a textbook, but you only had, 100 word set at a time. But if they overlap, you can align those together. And a scaffold would be the complete overlap of one section. And so you have a collection of scaffolds that maps to the genome.
Okay.
Does that make sense?
But maps, what do you mean maps to the genome?
Like you can align it to the genome.
But it
Like
is
you
the
have
genome. I'm
some-
being
some
intentionally stupid here. You're trying to get you to explain what you mean by mapping these to the genome, because that is the genome, right?
You're
No,
working out
this
where
could
it
be
fits in.
any sequencing prep, right? Like
Right,
this could be RNA-seq, this could be, this could be GROW-seq, it could be,
But what map
PRO-seq.
are you using?
You're using some sort of reference genome that is in a compressed format.
And that's what I'm talking about.
Oh, and?
And
what else?
the
follows
human
with that?
genome was used as a reference genome for the chimp one.
No,
Okay,
it wasn't.
look-
They did de novo assembly as well.
Which
For
they
used
used
it
earlier.
Like that
Yeah.
was from the
Yeah, we
initial.
provided the reference.
That's from
Yeah,
the
the-
initial reference.
The Nature 2005
Yeah,
one. Mhm.
like they did a de novo assembly, and then they used the human as a
Yeah.
human as a reference for the alignment, so that they could do interspecies comparison. That's not That's not unfamiliar. You have to pick one
Yeah.
species to do an alignment.
Yeah.
It's like this. It's like if you sequence from base pair 1 to base pair 100, right? And then you get another fragment that goes from 90, right, to 190, and then another one that goes from 180 to 280, right? And you keep going like that, and they all line up with one another, right? So now you've got this huge, long, contiguous stretch of sequencing. You're not using anything else. You're just using the sequence data that you have developed.
Yeah,
Right.
and if you have another set of fragments from the same genome, the same organism that are really big, so you have these little 100 base pair ones all stitched together, and then they stitch together on one that's maybe 1,000.
Right. So you do Illumina
That's the
sequencing, and then
the rest.
so you do Illumina sequencing, and then you also do Sanger sequencing, right? You do
Mhm.
other sequencing methods, and you get different sized chunks, but it's all just the DNA that you're starting with, and you line all of those up with one another, and then eventually you get the whole thing, right? And then you want to compare that whole thing to some other species. Well, guess what you've got to do first? You got to figure out how they map to one another. But that's only when you're doing the interspecies comparison.
We just sequenced the Sumatran rhinoceros genome here, and we did it by short Illumina reads. So we had some short reads that are 250 base pairs, and then those are scaffolded on longer fragments from the same individual that are between 2 kb to 10 or 15 kb. And we assemble all those together de novo, meaning we don't reference anything else to piece them together. That's a de novo sequence. And so you generated your genome sequence for that organism without any reference to anything else. And then you take that and then you compare it to everything.
And for the kids out there, don't ever take any classes on assembly. It's awful. Those algorithms are absolutely horrific, and they appeal to like.
I am
ancient
so old. When I hear
mathematical
assembly, I
crap,
think
dude.
computer code for some reason.
Oh, Sam, now you tell me.
I'm sorry, man. Just be okay with De Bruyne graphs. That's all
We're
I can say.
we're going on 2 hours. We're going on 2 hours. So what I kind of want to do is, if there's people in the live feed that have questions, if I can kind of open up to more of a Q&A.
Yeah, I
why
gotta
we're
go in
doing-
like 10 minutes, Steve. Thanks for having me on.
No, thank you, Sam. Appreciate it. I did want to touch on something we kind of jumped over real quick now that I got Isaiah here, because Isaiah hasn't joined in. Isaiah, do you want to touch a little bit on the cryptic centromere? Because you had posted something not too long ago on the fish hybridization that you showed very clearly that the cryptic alpha sequence is there. Did you want to touch on that and ask a little bit about that to Ben real quick, if you could? Isaiah, is your mic working? Uh-oh. You might have a technical problem. Okay, so if anyone, while we're waiting on him, does anyone in the live feed?
CRISPR has the fish stuff, does he not?
Oh, is that what he was referring to?
I'm wondering if
yeah.
Or was it Rational Mind that had the fish stuff?
Yeah, that's the paper, but he actually had a really good visual image, and that's just only the abstract, by the way. But
I thought CRISPR had that image as well.
Maybe, CRISPR, do you have that?
By the way,
the fish
for
image?
the audience,
Yes,
fish,
Should present it.
fish is fluorescent in situ hybridization, not
And
fish
no
as in
Isaiah can
swimmy
hear.
swim.
Yeah, yeah. Who was it? Who was it that actually thought it was talking about like Ronnie? Ah, hey, yeah, my good friend Ronnie, of course.
Yeah, that makes sense.
Yeah, that makes sense.
Oh, I also wanted to say I really would love to see some of those journals with computational analyses where you don't have to write like an extensive methods, because I want to publish at those journals and get the fuck out of here.
Yeah.
Only being
Yeah,
tongue in cheek. Sorry. about
Isaiah posted it. Can somebody,
You have to.
Rational? Can somebody screen share Rational's post, because he just showed Isaiah's
Try it.
post. That's what I was referring to. Yes, at the 2Q21.
I can't do it. I've gone all slow. Sorry.
Well, you know what? I can do it. What the heck? I can do it.
Well, while we're
No, I can't.
entertaining the audience with all of this dead air,
Yeah.
are there any questions in the live feed? I can't see them on my phone.
Let's have a look. Oh. How long does one of those things take to sequence?
The
Not-
minion.
Not the sequencing that takes a long time. It's the preparation of the sequencing
No,
library.
Yeah, he's talking
And
about
then the
the minion,
analysis.
the minion. How long does the minion take? I think it's 15 minutes.
The runs on a minion, you run to it as long as until you achieve the coverage that you want. So
Yeah.
the longest runs can be 48 hours.
Right. What's the read rate?
You have a smaller, if you have a smaller pieces that you're looking at, you could get enough coverage in 2 hours
Yeah.
to run. They use these in the field. They take them in the field to sequence the Ebola virus.
Yeah. What's the rate? What's the base rate?
Oh, the
How
read
many
rate?
bases per minute? Yeah. How many bases per minute?
I don't even. I mean, it's through the roof.
Well,
Yeah.
viral,
I could find it out. I just had a page on the minion not too long ago, and it gave all the.
Viral
I think they're
and
talking about
bacterial
like 3
sequencing is a little bit
gigabases in
different.
48 hours. So
It's
Have we
amazing.
got the screen share sorted yet?
Yeah, I'm actually presenting CRISPR now. He found the
Yeah.
what I was presenting right now. So yeah, see,
Yeah.
this is what I want to
Maybe-
ask about.
Yeah.
If
Heavy
Isaiah
Holler
can.
thinks it's fish PHISH. It's not. It's with an F. Fluorescent in situ
Yeah,
hybridization.
phishing is when you try to get somebody's password heavy.
Yeah. Right.
Okay, so
we
they added
know
a
that-
fluorescent protein to a marker or something.
Well, I was gonna have
Is that what
Isaiah
this
kind
is?
of go over this, but if you can't talk, then anybody else can. I wanted to kind of bring it.
I'm not seeing anything on the screen at all. It's just black.
Really?
It's from a jellyfish, right?
Let me try presenting them again. Okay, good. Now, can you guys see this?
No.
Really?
I've just got a black screen. CRISPR, are you screen sharing?
I can see it.
I can see it. Check your bandwidth, Fiona.
Oh, it probably is. Hold on.
Yeah, you may have to like turn it back up if you had it down.
I can see it.
And I don't think Isaiah's mic's working. So, CRISPR, why don't you go ahead and tell people what this actually is, what we're looking at for the fish? Or not. All right. Isaiah says on the side, Baldini cloned a roughly 1 kilobyte sequence of alphoid DNA and found it only hybridized with ape centromeres. It also hybridized with the punitive centromere Mike. Oh, sorry. With this Mike not working had nothing to do with the Baldini cloning. They said it also hybridized with the punitive centromere. So if you look at the 2q21, that actually would be the cryptic centromere, Ben.
How long is the alphoid Oh, that's a 1K sequence. What I meant was how long is the working human centromere?
I don't know base pair. How he wants to make base pair of the working centromere.
It's several tens of thousands, I want to say.
Well, he said.
CRISPR, are you there?
Well, actually, Isaiah's in the side thing here in his own post. Says it's about 171 base pairs.
Yeah, CRISPR. Have you got the explanation for that? The fish data, the image.
Well, I mean, I've got the fairly long paragraph that Isaiah had typed up.
Yeah. Explaining why you end up with the two colors on the human chromosome.
No, stop moving it. I'm trying to read.
Sorry, my internet's pretty slow right now too. So.
Right, we've got two probes. We have a probe that is specific for the centromere on the ape chromosome. And we have another probe that's specific for the centromere on the human chromosome. And when you use those two probes together against the human chromosome, both of them bind. And you can tell both have bound because each one is labelled with a different fluorescent dye. So you get, I think it was a green spot and a red spot. One is where the human centromere probe bound, and one is where the ape centromere probe bound. So they're finding the ape centromere.
Is the 2q13 that's the fusion site, right? Because there's like 213.
I couldn't tell you. I mean, I'm doing this off the top of my head.
Well, it would
because I can't. I'm doing it off the top of my head because I can't see it.
It would be two human centromeres. It's not a fusion of an ape chromosome with a human chromosome. It's two human centromeres is the idea.
What?
I don't know what you mean by that either. I mean, humans are apes.
How many?
What do you mean by apes?
No, it hybridizes with the ape-specific centromere probe.
Oh, the alphoid sequence that's being detected on the ape probe. That's right. Okay.
Which is the two kind of what is that? I don't know what color that is. Brownish.
Sorry?
You can't see, but I'm trying to look at the color on there. It's kind of like a brownish color.
I have no idea what you're looking at, so I can't see.
I can't
see anything.
You want to? I can give you the actual link. You can pull up, locally. So here.
That's what we're going to do.
Yeah, it's right there.
Is that the one rational? Right, hold on.
Okay.
2q13 is it? Disappeared.
Oh, sorry.
2q13 fusion site.
Yeah, that's the fusion site.
Is the cryptic centromere, and the red one is the actual functioning centromere. Yeah. That looks fairly similar.
That's the diagram I was expecting.
And when we were saying before that the one before with the banding just showed there was a banding thing there, that showed a relationship. Then when they actually did the fish and they did the actual sequencing, they've actually determined that it is a cryptic centromere.
Are these the 2 human chromosomes or one ape next to a human, or you're just saying it's binding to the?
I've lost that again.
That's the human number 2.
So that's okay. But so this is not a comparison of the 2.
I think it's just looking at from different.
Human
No,
number
it's
2.
showing you what happens when you use the probe.
Yeah, he says those are the 2 human chromosomes. So this is not relevant to the diagram.
What?
What diagram?
We're talking about the human chromosome.
I thought you were talking about
the human. We're talking about the human chromosome. How is a diagram of the human chromosome not relevant to discussion with the human chromosome?
I thought you were talking. I thought you said the diagram that Tomkins wrote about.
We're just pointing out that it's claimed that they haven't found a cryptic centromere. That there is 2 centromeres here, and it's the only chromosome that has 2 centromeres. That they have found a separate centromere exactly where it should be if there was a fusion.
It's not a centromere. It's a 171 base sequence called an alphoid that happens to be the same.
Alphoid DNA is in a centromere.
It is also in other places.
Right.
And it
Well,
binds.
where are the fish sites bind? If it's in other places in the chromosome, where are the other fish sites lighting up?
Well, see, Isaiah says chromosome 9 also has 2 centromeres because according to alphoid sequences.
Give up. I just give up.
Well, what are you trying to accomplish? I'm curious. You can. This is an honest question, also.
The hypothesis is human chromosome resulted from a fusion event of 2 ape chromosomes, so it should have 2 sites that light up the centromeres.
And you accuse creationists of confusing evolution. It's the common ancestor.
I'll tell you what I'm trying to achieve.
Humans.
I'm trying to achieve somebody getting to finish a sentence.
And I don't know why I keep bringing up creation, evolution. None of us are bringing up creationism or evolution. None of us have even mentioned either of the words, I don't think.
Well,
this has to do with the actual claim that this is actually a fusion. And what we're trying to determine, is it a fusion or not?
The reason the fusion has been hypothesized is to explain why humans have a different number of
But the evidence is the
chromosomes than
evidence, right?
chimps.
We're just looking at the evidence.
You
We're actually
found
looking at
the evidence because you were looking for it. Okay.
Excuse me, the evidence that could actually, you could even argue. Stop laughing. May I finish?
Go ahead, Fiona.
Here's the point. That chromosome 2 fusion can even be used as an argument for young Earth creationism.
Or
You
intelligent
don't
design.
get that, do you? This is the point.
I do get that.
And he does it again. Then, if you do get that the fusion could also be said to be evidence for young Earth creationism, why are you going on about creationists and evolutionists?
What?
In other words, this evidence isn't solely just evidence for like common descent. Matter of fact, Ruhev actually is the one that actually claims nobody should be using this for evidence for common descent for that very reason, because it could be used. Anybody could look at it and go, you know what? I want to make a model for this that it supports the young Earth hypothesis.
What you're saying is that the human chromosome could have fused, and that does not prove that there's an independent, or that does not show a common ancestor between chimps and apes. Right?
Yes, exactly.
But
exactly. That is what I've just said.
Mapping it to the chimp chromosome is an explicit attempt to show where the 2 are different.
You're getting
it. The point is that you can argue that God did it that way.
Right, in situ, he just kind of made it like that. Proof.
That's bullcrap.
Let me finish, please. You've interrupted me every time I've spoken now, please. You can make the argument that when you look at genetic similarities right across the animal kingdom, that's because it's a common creator. He used what he had already made to make the next thing. Mm-hmm. And that argument has been used by creationists and by intelligent design advocates for years. And therefore, that same argument that they are making can also be made for the human chromosome 2.
Can I give my preferred So this is my preferred young Earth creationist explanation for it, right? So as Fiona was saying, common design, right? So that's why you've got it packaged the same way. Created with humans originally, 48 chromosomes, just like the other apes. The fall happens. Everything starts getting more corrupt. The fusion occurs, right? Just as we see. Very low population at the time, easily fixed. There you go. Perfectly consistent.
And I've actually people have done that. That's
I
not.
don't believe that is the case, and
Well,
I
right,
don't.
that's what Tomkins argued too.
You may not believe it's the case, but what I'm pointing out to you is the fact that you keep talking about creationists and evolutionists, but it's got nothing to do with whether chromosome 2 is fused or not.
It
It's
really does.
irrelevant.
It absolutely does. The reason this was proposed was to explain why humans have 46 chromosomes and chimps have
Yes, and
48
that's the
and
point.
still have
It
a common
may-
ancestor.
Yes, and that may be the explanation, but there are other explanations, and this is what you're not getting. There are other explanations.
Can I just say that I disagree with the way that you're framing this? It was not proposed to answer that question. That was an open question. They found, hey, look at this. These two chromosomes, if you put them end to end, right, they line up really nicely with human chromosome 2, right? That answers the question. They weren't trying to answer that question. They weren't, right? It was just that is the case. Therefore,
Yeah, when they started doing the banding, it's kind of hard not to notice the similarities between the chromosomes when you look at the banding. I mean, it's just spot on. I mean, so when they found the banding thing, and they didn't have the technology, correct me if I'm wrong, but they didn't have the technology at the time to really get more information to see if this is actually what it was. And it wasn't until they did it was in the 80s, it was in the late 80s, correct? Mm-hmm.
Yeah.
It is quite possible that there was a fusion there. I don't think that's the case, but for instance, creationists think that all dogs, for instance, descended from the same basic kind. And there are different kinds of dogs and foxes and
I don't want to get
wolves
into the
that have
kinds thing.
different chromosome numbers, is what I'm saying.
But let me ask
so
you then, have you looked at the nucleotide sequence that's actually at the fusion site? If anybody has that available, the actual the telomeric repeats.
This is what you told, yeah.
So, Mike, so you have seen it, right? TTA.
I'm lagging too badly.
There's not all of them are pristine, but for the most part, you can tell that they're telomeric repeats, and then the converse, the telomeric repeats. If it wasn't a fusion there, what would we expect to see other than that head-to-head alignment?
Well, what you want to ask the question is, what is the function of that alignment? And I think that there is a possibility that it folds into a stem loop structure.
Well, I'm not asking about the functionality. I'm actually about the sequence itself.
The sequence and function are related. The sequence is not a palindrome, but it's got the sequence of the 6 nucleotide sequence in the telomere repeat, and also the complementary sequence right next to it, right?
Quite a few of them.
also, can I just
And if it's complementary, that the meaning of complementary sequence is that it binds to each other in a normal double helix alignment.
No, this is the
But
same strand.
if it's on the same strand,
Also, can I just point out really quickly, when you guys said that about the 80s, actually the evidence was actually originally 1973.
Yeah, that's true, actually.
It actually predates the Unison Prakash paper. So,
and the point of it being complementary is because with a head-to-head fusion, then one of them will be, as we could call it, upside down, i.e. complementary on the same strand.
mm-hmm.
So what are we looking at, Ben?
Think
Here
about the geography of that.
If this isn't a fusion, what would be different here?
I don't understand the way you phrase the question. What I'm trying to say is that's the sequence, and here's a possible function of that sequence. Does that make sense? And why it's complementary to the side on the part right next to it? Because it could form a stem loop structure. We know RNA does it, and we're starting to find out that DNA, the same strand, binds itself when it's unwound. And it affects
DNA's double-stranded. It's already.
But you have
So-
to unwind it to transcribe something, and that's what
No,
actually
you have to do more than unwind it. You have to actually unzip the two strands. It's
That's
double
what I mean.
stranded.
I'm still,
And the
yeah, I'm
double-
still. How does this happen on one strand? Is what I'm at. I don't know what you're saying there, Ben. If this is on a single strand, how do we get the complementary opposite reverse motifs here on the same strand? How does that happen with the double helix? Even if it just unzips, it's not going to do that.
Exactly.
If it unzips, it's a single strand, correct?
Yes.
And
Yes,
it's
and that-
being,
And that can form single strands can form a stem
and
loop.
part of the way that DNA works has to do with the three-dimensional folding that are far beyond in terms of
Of the
levels
double-stranded
above
helix,
just
yes.
the double helix. So there's all kinds of folding that's involved.
Well, this
And
would
part
have to
of
be
the
this
way
would have to be cut and then ligase together, though. Correct?
No, meaning it has
No,
a
wouldn't have to be.
it has a function that
No.
it involves the complementary binding of the same strand after it's
Do you have
unwound.
evidence for this function?
No, I'm spitballing.
What evidence do we have? That
But this is the
double-stranded
sort of
DNA
questions that scientists want to
Oh.
investigate.
Has anybody?
What evidence do we have that double-stranded DNA, when it comes apart from transcription, that the single strands then form stems and loops?
My perspective is that everything has a
What evidence
function.
do we
That's what
have?
I'm going to
What
assume.
evidence do we have? What evidence do we have that DNA, when it unzips to be transcribed, forms a stem and loop on the single strand?
A sequence is complementary.
So has anybody found the stem and loop?
You find it in RNA, and I have. See,
Well,
Isaiah
RNA
here
is a
is
single-
Isaiah here knows what I'm talking about. He's calling
Please.
it a palindromic sequence.
And I know what he's talking about as well, but RNA is a single-stranded molecule, not double-stranded. Double-stranded DNA isn't going to form that stem and loop
Not when
in the
it's
same way.
not when it's in a double helix form. That's correct. But in order
Exactly.
to transcribe something, you have to unzip it, as you know, and as you
Uh-huh.
said. So it will be in a
And the-
single
and-
strand at some point.
Yeah, and if a stem
And
and loop
that-
forms, how is it going to be transcribed?
That's not the part of the gene that's being transcribed. It's an intron.
Right?
It's a promoter sequence.
Oh,
It's-
for
it's-
goodness.
So it's not unraveled to form a stem loop
Oh.
because it's not transcribed, right?
It's a binding site for transcription factor.
I don't even know whether that's critical. Why does it matter that there's a transcription factor binding site there, anyways? I mean, they're ubiquitous all over the place.
Yeah,
right?
they're binding everywhere.
Because everywhere in the gene probably has a function.
Yeah.
That's an
That's a-
assertion with no
Probably.
evidence.
Probably.
And that's what Tom.
Well,
that's,
there's
that's what
not-
I think, I
there's
think that's
not
what the
go-
criticism of Tomkins is right now, though. If you know that
Exactly.
he's saying there's a function there, but you can't just say there's a function there without giving what
Like-
the function is.
Like I tried to say earlier, this is going to be a difference between the way creationists approach
no,
this
I think
kind
this
of
is
question.
science. Why would
No,
this?
it's not.
Why
It's
is
just
it
you
have to do
approach it
creationism?
you approach it with bias.
I think honestly, you're
We're
showing
biased
a little more
towards
confirmation bias.
towards believing that there
You
is
assume-
function
you assume that
there.
there's function
That's what
everywhere,
we're
which
biased
is- I don't assume that there's not function everywhere. I don't assume that there is function everywhere. I assume that there may or may not be function everywhere, and I need to go find out whether or not there is. And there are ways to do that, and I can do them, and so I do. That is the way you do
Yeah.
science. You don't go, oh, well, I assume that this must be functional because everything is functional, and therefore I'm going to keep looking at it until I eventually find some kind of thing that I can call a function.
Yeah, that's confirmation bias at its
Yeah.
finest. I mean, that's you can't have an objective point of view and work in a lab like that. I don't think.
What Isaiah said, the CTCF value, there are several more transcription factors that he reported in his paper. That's just the one Ruhif decided to look at because the factor was so low.
What is wrong with Isaiah's mic? Out of curiosity.
So he was looking at several transcription factors, not just CTCF.
Do we have any more questions from the live feed? And does anybody else want to ask questions
I'll
about?
look. I'll look.
All
I'm
right.
going to mute myself.
Thank you. If you want to take care of the live feed like you usually do, that'd be awesome. Is there anything in Tomkins' paper that people want to ask specifically? We got to the transcription levels, we got to the expression, we got to the cryptic centromere. Is there anything else in here that people want to ask about? I know there's a whole part on co-expression. Unfortunately, Sam's no longer with us. He's probably the best to talk about when it comes to co-expressed genes. But I don't even know Tomkins' real argument when he's actually using co-expression to determine functionality.
Well, it means they're part of the same cascade of gene regulatory networks. So if something, if
No, it means they're expressed at the same time.
Yeah,
which is not coincidence. It's a result of.
No, you assume it's not coincidence.
Yeah, that's the problem. I think it has to be showed that it's expressed.
I would agree that it has to be showed, but I think it's a good assumption and a good hypothesis that they're co-expressed because they're part of a similar gene regulatory network.
Actually,
that activates them both at the same time.
Sam's lab has just put forward some work showing that it usually doesn't indicate that. But again, we really would need him in here to talk about it. But
you have one of his papers. I could look at that. That sounds interesting.
I don't have that to hand. But
what's his name? I can maybe look it up some other time.
I don't think he was listed on that paper, but it's not really the point.
And what am I looking at for this thing you just posted there?
Well, if I'm supposed to use the same standard of evidence that you guys are, I would need to ask for a published paper on that, and if I didn't get it, I would just laugh at you and say you don't know anything.
Oh no! Wait a minute. That's not what people have done. That's not what people have done.
Yeah, we're addressing the claim. I mean, this is Tomkins' own claim. So I mean, if he's just making assertions, that's one thing. But he's making a claim.
And an argument was presented that co-expression doesn't mean anything, and no evidence was given.
That's not what
and so.
No, please. If we could just maybe stop with the interrupting.
Yeah, they didn't say co-expression doesn't mean anything. I think the argument is that co-expression really doesn't tell you, give you certainty that there's expression across the board with the things in the same network.
Science isn't about certainty. It's evidence.
Co-expression tells you that things are expressed at the same time, right? That does mean something. It means so. If you want to say that gene A interacts with gene B, right? That can't happen if they're not co-expressed. Okay, so you need to, if you can show that those two things are co-expressed, well then it's possible that gene A interacts with gene B.
But you have to show that A and B are expressed,
right?
Period. Though, and if he's saying that this is expressed and it's not, then why was it saying co-expression matter then?
What I'm saying is that co-expression says that if these two things are co-expressed, then they may interact with each other. But now you have to show that they interact with each other. It's not enough to just say, oh yeah, they're co-expressed.
I understand that, but it is evidence.
It's evidence that they're co-expressed.
Yeah.
It's evidence that there's possibility that they're interacting with each other.
Well,
and I think you just explained that very well.
I think he's going beyond possibility.
No,
he's actually making a confirmational claim.
Please. The fact that the two things are transcribed at all is evidence that they maybe could, possibly, who knows, interact with each other. You don't need co-expression for that. In fact, you could make up two random sequences and say it's possible that somewhere there exists an organism that expresses these two sequences and they interact with each other. Sure, that's possible. But now what you have to do is show there is an organism that has these two sequences, they are expressed at the same time, and they do interact.
And that part is really, really hard, and it's expensive, and it takes a long time to show that biochemistry.
Not really.
Yeah, it's a lot harder than accessing.
But Ben, isn't Tomkins' argument that it is highly expressed? Like, not that it's possible. Not that he's saying that, hey, these things are co-expressed, therefore it might be possible that there's an expression here. He's saying, if I'm reading the co-expression thing right, that he's saying because of this, it is highly expressed. That's a different claim altogether.
The gene is highly expressed.
But it's not.
Well, yeah, that's what is usually expressed.
You said that the evidence you presented argued that the long variant is not highly expressed.
And that's what he's trying to show negates the fusion, though.
Yeah.
That's the whole point of the argument. He's showing that long alternate hypothetical transcript to
He's
be
not
refuting the human chromosome 2 fusion, and it doesn't do that because there's no evidence of an expression of that alternate transcript.
He's not arguing that because it's highly expressed, it has a function. He's arguing that it's likely it has a function because it's co-expressed.
The problem is that he says it is highly expressed, and
in the
it is
paper.
not.
Within a gene.
How can you work out the function when said transcript hasn't been found to be expressed?
It has been found to be expressed.
No,
it's very low.
No, it's zero. It's zero.
CRISPR showed you the bar graph showing that it's not.
The data that he's looking at is also co-expression and things like that, and that's legitimate expression.
CRISPR, can you remind me that bar graph that you
It was
showed?
at 0.
Yep. And
Also
was that
co-expression. Co-expression
was
does
that-
not automatically imply function. Expression does
Yeah,
not
and was-
automatically imply function.
Yeah. And wasn't that bar graph that you showed that shows no expression, or one of the links that Tomkins gave claiming high levels of expression?
I don't know if he links to GTExt or not.
I think he did. That rings a bell for me.
GTExt is sort of the gold standard for this anymore, so
Yeah.
it's just... If he
Yeah, I mean,
wasn't
I
using it,
think-
he should have been.
Yeah, and that I think is a problem, that you cannot say that they're co-expressed if one of them isn't expressed. To be expressed, you should be finding a transcript, because remember, you're talking about a long transcript.
Also, can I just say that if you have two things that are both expressed at background levels, right, then they will automatically, by the very nature of what co-expression is, they will automatically be co-expressed with each other, right? Because they're both at background.
Exactly.
So they're always at the same level in the same places because they're both at background.
And why would an organism do that? Have expression levels that are at a background level that's very low, and
Because
that only ramp up in
background-
specific
background
situations? I understand what
you
background
don't have
means-
evidence that happens, for one thing. Second, you have to understand that transcription factors can just, a transcription factor binding site is not exactly that big, and they're pretty common, right? So, for instance, the telomere repeat, right, is about one mutation away from being about 8 or 9 different binding sites. Okay? So I mean, that should tell you something right there.
Well, it's actually interesting. It does tell me something, and that's something that Tomkins was talking about, is there's many different transcription factors that are possible binding sites there.
Right, but you understand my point is that
Well-
if it's so easy for there to be a binding site, the fact that there is a binding site isn't surprising.
It's not easy.
Well, no. Well,
It
he's saying that
requires design.
if there's so many transcription factor binding sites all over the place, what makes this one meaningful in any way?
Do you remember me saying that I ran a 1,000 base pair random sequence through a motif search engine, and it found several hundred? Okay, that does not indicate that it's difficult to have one of these things happen. It's not like they're 30 base pair long or something, right? They're 5 or 6 base pair.
I thought you would say that a longer sequence would be more... A shorter sequence is far less complex and more likely to be randomly...
What?
Yeah.
What?
That's the way binding site motifs work.
Yes.
They are short.
Mhm.
A transcription factor binding site is a short motif.
Right.
It occurs
So-
randomly, very readily.
As part of gene regulatory networks, usually.
No, I'm saying
Randomly,
that
for no reason.
Yeah. What I'm saying is, would you agree that if I produced 1,000 base pair random sequence of DNA, right, and I inserted it into an organism, right, that thing, I have no intended function of it because it's random, right?
Sure.
Okay. Do you understand that it would have randomly a lot of binding sites?
Because they're very short sequences.
Yes. Does the fact that something binds there mean that it has a function?
I think it's evidence that it could, and I
I
think
just told you it's a random sequence.
It doesn't have a function by definition.
Okay, so here's another argument that we haven't talked about yet.
Functionality. What
If
constitutes functionality?
according to evolution, okay, and they've looked at this. There's lots of papers on this type of phenomenon. When something is non-functional in the genome, it gets deleted out within a few generations. Why? Because it's not.
What?
What?
Not fairly.
I'm not. I don't want to straw man you. So can you please repeat that again, though?
Things that are not functional eventually get lost in a population out of the genetic information.
No.
Because, yes, it does. And we know this from bacterial studies where we...
Yeah, in bacteria,
From
maybe.
bacteria, that's hugely
Humans-
different.
You just
are
told
not
me
bacteria.
no.
But we're not bacteria.
We've been talking about humans.
We're talking about human beings.
It
that's
should
what
happen anywhere for the same reason.
No.
No.
It happens in bacteria because they're under intense competition with one another to replicate their DNA.
And they
fast as
already
possible.
have very, very small genomes. Right?
Right.
Yes.
So they're under intense selection. Lose those non-functional genes.
So are
We
humans
aren't under that same selection?
Yeah. So I
We're
mean,
not
between the
under
small
intense
genome and the fact
selection?
that their genome changes constantly.
Humans are barely under any selection anymore.
Yeah. But
Well,
Yeah.
but over millions of years of evolution, that's the relevant
We're
time.
not under the same selection. We aren't under the same selection as bacteria are. It's just they're different organisms, different environments.
So now we're under the same selection as bacteria. When you
I didn't say
Yeah, if we don't survive, we don't leave offspring. That's the
bacterial
point.
genomes are under a lot of selection to replicate their genome as fast as possible. That means they want less DNA. We don't have the same selection to replicate our genes as fast as possible because we're multicellular organisms. Our
It's-
cells aren't in competition with one another
It's the
to replicate
same.
themselves as fast as possible. So
It's
they
the
can
s-
keep pieces of DNA that are non-functional. Non-functional DNA doesn't just disappear because of evolution.
Or more
It's
to the
not
point,
under that strong a selection because we're multicellular.
more to the point, the function of the genes plays a much, much higher impact in the selective pressure than the amount of slack. So for a bacteria that has a fairly small genome, right, any extra slack in the genome is going to be a detriment, a fairly substantial one, because they have a short lifespan, and any hit matters, right? But for a big, complicated organism, other things matter a lot more than whether or not it takes a few more nucleotides to complete your genome, right? That, in the scheme of things, for a eukaryote, that's just irrelevant.
Right.
Ben, do you suggest like the whole human genome has functionality, like every?
No,
Or-
I'm open. I mean, obviously, I would say that the original design we would assume was all completely functional in some way, even if it's just a spacer in the three-dimensional structure so that certain genes were in the right place. You know, it may not even be transcribed at all, but it serves as you know, this is the part that's inside a nucleosome, not exposed, and here's the part that's outside. But so basically, I would say that all of it originally had function,
So you're
and that it lost function through mutation. And if there are non-working genes and non-functional parts of the genome, it's because it lost the original function.
Ben, can I ask you a question?
Oh, that's interesting.
Can I ask a quick question? I just need to know this. When you say the genome originally had all this function, okay, is that something that you're saying because of your religious convictions?
That's part of creation science theory.
So, but in science, we don't start off with a religious conviction like that as a hypothesis. So when you say,
That's not a
well,
religious
our genome
conviction,
started
that's a
off with all this
historical-
function, let me finish real quick. When you say our genome started off with all this function, that's not something we as scientists can just assume, because we're not obliged to share that religious belief.
I'm not trying to make you share a religious belief.
No, but
That's
that's where your basis. That's where your basis for creating this hypothesis is coming from. You're saying the genome is functional or was functional because
I'm trying to evangelize you?
No, that's not what he's saying. You're not understanding what he's saying, Ben. Ben,
why you think that.
What he's saying, Ben, is that hang on, Ben. What he's saying, real simply, is that your position that you're coming from is based upon your need to satisfy your religious beliefs, rather than what the science is actually indicating or how science is actually gone about. You're starting with your own presuppositional ideas.
We're starting with historical evidence.
What
From,
do you mean?
From the Bible.
But that's not how science is done.
It is done that way in certain instances. For instance, sometimes you use ancient volcanic eruptions like Mount Vesuvius to calibrate radiocarbon dating or other kinds of dating. They do that, and they base it on historical evidence of where and when these events occurred.
Well,
it can be used.
Are you saying like Adam and Eve is historical evidence that can be used scientifically speaking?
Yes, I'm a young Earth creationist. I believe that Genesis is an accurate historical record.
My question is scientifically speaking.
You're trying in this discussion to convince us of a scientific conclusion.
I don't know what
scientific conclusions sometimes reference historical evidence, and another example would be in the global warming debate. We can open up another can of worms here. So
we don't reference historical data based on a religious belief in religious text. In climate science, you're asking us to buy your argument. We have to buy in your belief in the Bible. And that's not the way science works.
In order to participate in secular academia, I would have to buy into all kinds of assumptions that you guys make.
Because if you can disprove any of those assumptions, you
can't disprove assumptions. That's the nature of an assumption.
You can validate
an assumption. An assumption is something that you assume to be true that helps you.
It's not provable, but you can definitely falsify things.
Yeah.
You can falsify an axiomatic assumption. If I take the assumption that decay rates haven't changed, and then I could develop a method to show that decay rates have changed, I have then invalidated that assumption.
That would have to be based on historical evidence. Though.
I assume that the sun's going. I assume that the sun's going to rise tomorrow. If it doesn't, then I'm wrong.
Look, when you guys reference a paper that was from 1973 or whatever, that's a historical record of something that people back then saw and recorded and put down. Okay. Now, I'm not saying the Bible is a scientific manuscript. That's not what I'm saying. But it is a historical.
But you're invoking a supernatural mechanism.
No, I'm not. I'm invoking a text that has been found, that was written by people, that claims to have a history of the world.
From supernatural origins.
Some of it potentially, but
why that text? Why the Judeo-Christian text? There are hundreds of thousands of texts written. Why don't we refer to those?
We have
Could
lots
I just
of
address
evidence.
Something that Ben said, please?
Yeah, go ahead, Phil.
Yeah, go.
Ben said something about, if we look at a scientific paper from the 1970s, and that'll be a historical document and all the rest of it. But here's the difference. We can look at the methods section of that paper. And we can replicate their work and see if the conclusions hold.
Right, and that's one problem with historical and origin science is some of these are one-time events that you can't really replicate in the lab.
I think that Jesus forgot to include his method section,
including evolutionary events that have been proposed as one-time, non-reproducible events, like a human chromosome fusion.
Well, that's why
you can't reproduce the exact fusion of the human chromosome.
We're not trying to.
Funnily enough, though, we do have living examples of humans with 44 chromosomes, and they're perfectly normal. There's a guy in China, he has 44 chromosomes. There is also a family in Finland that for 9 generations There have been
Interesting.
individuals with 44 chromosomes.
We also have examples
And
of
they are-
elephant
and they are-
man and. Sorry, go ahead.
Please.
I thought you were
just-
done. There's some lag here. Okay, I apologize.
So here are these people. They are short on their chromosomes, but they have the full complement of genes. They haven't lost a chromosome or two. There's been a fusion. So they have all of the genes that a 46 chromosome human would have because of a chromosome fusion. And these people are normal. You can go and look these cases up.
Sorry, I think
I
that
believe you.
the common thing in creationism that you say, well, we can't replicate this fusion event that occurred, so we can't prove that, that's a typical creationist argument. However, that's not what we're trying to do. We see the data, we see these sequences and chromosomes, banding patterns, all this stuff, and we look for hypotheses to explain those data.
Right,
So,
and that's perfectly illegitimate. I'm not criticizing
Finish,
that.
please.
The best available hypothesis we have to explain the available evidence is that event in the past in an ancestor to humans after they diverged from apes, from chimpanzees. That's an explanation. We don't have to go and repeat it or go at a time machine or anything like that. That's how virtually all of science works that way. Alternative hypotheses and finding which one explains the evidence best.
I would
And
say,
make predictions from that.
am I being asked questions or you just talking at me?
No,
Go ahead.
There's interaction going on. We're just saying that the repeatability has to do with experimentation. You don't repeat one-time events, you explain them. That's what science does. It explains what happened.
Well, usually what you see is you're trying to work out something that you have that's in front of you. Like, for instance, if you're trying to cure cancer. Okay, it's there, you have cancer cells, you have human beings that you're trying to help, and it's there, and you can see what works, and you can see what doesn't, do clinical trials and all kinds of things. Historical origins science is a little different on both sides, not just creationists or evolutionists. We're both trying to explain historical events based on evidence we can see now. But the events themselves are not repeatable, not in the same way as...
Well, they're not, but that doesn't matter. They're not repeatable.
It does matter.
No, it doesn't. The Big Bang, not to go to another rabbit hole, but like Big Bang cosmology, we don't have to repeat the Big Bang in order for us to have a model that describes what we're observing today with the cosmic background background.
To have a higher level, to have the same level of
let me try
confidence that it was correct. Okay, you as modern science, which is what everybody sends all this money to you for, because they know that the method works to help us, increase technology, increase standard of living, fight disease and things like that, because those are all things that exist now. And the research that you do on those can be done and repeated in the lab, in populations now.
What we do is we repeat we see things. For example, we had mentioned to you, Isaiah. Obviously, Fiona had said about the chromosome 44 chromosomal man. You kind of tossed over it because that actually shows that these telomeric fusions do exist. These ribosomal translocations can happen without detriment, side effects, without ill effects. I mean, there are some side effects when it comes to the offspring, but
telomeric fusion.
Did
they sequence those guys?
Steve, can I try something? Please?
I would.
Oh, please.
What are you trying to do?
let me.
I asked that a while ago, and
Ben,
I'm not sure.
let me ask you something here. Imagine that an organism underwent a telomere to telomere chromosomal fusion. Okay, what do you think it would look like?
I think it would have a very long telomeric repeat. I think it would have a very long subtelomeric region, which is, in Tomkins' paper, he says it has, 100,000 bases or something, a subtelomeric region, and it would all look the way it would have before it was fused.
Wait a minute, hold on.
What we have is an 800 base site.
Let him finish, please. No, just let him finish, please.
Yeah.
What we have is an 800 base site with a few areas here and there that may map or may not, right in the middle of what could be and what looks like a functioning gene. It's not an.
So is it fair for me to say that the entirety of what you would expect is full-length subtelomeric and full-length telomeric regions end to end?
Directly after the fusion, yeah. And in evolutionary terms, the amount of time that has passed, as you guys were arguing
I'm just
very
trying to-
well earlier.
I'm just trying to completely lay out. I'm just trying to completely lay out what you would expect, not what we see or what you wouldn't expect or anything like that. Just everything that you would expect to see. Is that it?
You would potentially see. Well, okay, yeah. Obviously, you have somewhere you're going with this, so go ahead.
Okay, so so you would expect to see full-length telomeres.
Subtelomeres.
I'm sorry. I thought you said you would expect to
telomeres
see
and you said
full-length
subtelomeres.
telomeres and subtelomeres.
I think that the telomere sequence that would be left would be a lot longer than 800 bases. Perhaps not the full
Why?
length.
You would so you think there'd be long and pristine?
It depends
Can
on
I
how
just
far
give
long
an
ago
example
it
here,
happened. Whether it's
Please,
pristine or not.
please, actually don't, Fiona. I'm trying to get him down this, and it's just, it's just.
Okay.
Okay, so the thing is that you can do repeatably experiments in human cells where you induce fusion events, right? Telomere to telomere fusion events.
Are these cancer cells you're talking about, or just any?
They don't have to be cancer cells.
Are you referring to a particular study, or just?
Yeah, there have been a couple of studies. You can also do this with yeast, by the way, which are not cancer cells, importantly. And the point is that on average, you see very, very few repeats left. Sometimes none.
Okay, well, show me the study. That sounds interesting.
Well, let me ask Ben. What? Okay, if these were, how would a telomeric fusion occur if these were like pristine telomeres and the repeats were intact? I mean, why would you have a fusion to begin with?
Yeah, you could not have a fusion if
That's
the
why
telomere is full length.
That's why I said subtelomeric region, if you were listening, and I did not say. Said I thought there would be probably a lot longer telomeres than 800 bases. I did not say the full length.
Is it possible, in fact, that the entirety of the telomeric repeat is gone?
Maybe. I mean, anything's possible.
Is it possible that, in fact, it's chewed into the subtelomere somewhat?
Right, and this site is supposed to be in the subtelomere. I know that. That's the
That's correct,
location
yes.
of it. Well, show me the studies you're referring to, and that sounds interesting. But
So-
you know,
so you would agree
another thing that is an issue here is, from a young Earth perspective, we could say, for instance, that Adam or Noah or one of them had this fusion event. Okay, and that's one individual had the fusion event, wherefrom we're all descended from them. But under an evolutionary perspective, you have a population of people that you start with, or a population of pre-human hominids, whatever, whenever this happened, okay?
But Ben, hang on one second.
I'm not done yet.
But that's not the argument, Ben.
In order for the event to propagate through the population, it has to have a positive selective effect of positive selection.
Ben, we're not arguing evolution versus creationism. We're arguing, is it a fusion or not? Right? That's it. We're not arguing Adam and Eve. We're not arguing if it happened 6 million years ago. We're arguing, is it a fusion site?
It's a fusion site that exists in all human DNA today, except
but
for those.
Tomkins is arguing that it's not a fusion,
Steve,
and he's
just to be fair, I'm not arguing that it is a fusion site. I'm arguing that it looks like a fusion site would.
oh, fair enough. Right. Okay. Yeah.
Oh, I don't
So-
doubt that it
so-
looks like something you would think a fusion site looks like. We would doubt that because of the fact that
But you've just
functional elements are not likely to be randomly assembled by something as massive as a fusion event.
so explain our Finnish family.
Which is a fusion event.
And
Yeah.
if you have a sequence, we can look at that and see if there were, functional sites that are joined at the fusion site.
Well, the point is that these people are perfectly normal.
And they have 44 chromosomes.
So
Yeah.
they fused at a spot that wasn't functional. I guess that would be another thing I would expect, is that the fusion site would not have function around it other than...
Can I use my handy teaching
Yeah,
aid
go ahead, Fiona.
just to make sure
Yeah,
that we've
but
got
not-
this sorted out? Okay.
Yeah.
Here we go. Chromosomes. Okay. The little blades up the top, right? This is the top of our chromosome. Telomere on the end, and the little blunt bottom bits. Wee bit there. Now, so here's the head. When they fuse in a head-to-head fusion, remember, we'll do it that way.
Well,
Telomere.
that's not the argument. The
Telomere.
argument is that the telomeres were degraded because the function of telomeres prevents fusion events normally. And in order for a
that's
fusion...
irrelevant to what I'm showing you.
Well, look, you other guys need to support me here when this happens, okay? If you have a fusion... The telomeres, the purpose of them is to prevent that sort of thing. And so they would have to be degraded significantly in order
And in
for
the
it to
original
happen.
paper, it said that they were degraded. In the paper from oh my goodness, who wrote it?
So-
Wasn't Costa.
So
but they said the region was degraded, and then and Tomkins make a point to saying, well, it's a degraded region. Well, even the original paper that demonstrated that it is a chromosomal fusion said that it's a degraded site.
Right.
Actually, in the paper.
I'm just making sure that you guys understand the hypothesis here.
Yes, we do.
Because it's a highly degraded region. But I would
No.
expect it to have more... You're asking me what I expect. I would expect it to be highly degraded, but to have more than an 800 base sequence of telomeres.
Why?
That's all.
Why would there be a limit to the amount of degradation that it underwent?
Right. Why?
Well, probably because the sequence around is so highly degraded that why would it have preserved the exact fusion site and not all this other stuff?
But it's not pristinely preserved. That's the point.
And it
The
did.
fusion
preserve-
site, you're claiming the fusion site is
Remember-
pristinely preserved. You say you know exactly the base pair where it happened.
But the
But
repeats are not
what-
preserved. Sorry, go ahead.
What I'm
Fiona.
saying is that these ends would have had to have undergone degradation. They've had to get blunted, okay? That's the telomeres. Nothing else underneath them, right? And that would then allow that to happen. And so you end up with two shortened telomeres. And it'll also mean, if you imagine that here is the centromere for each chromosome, you end up with that, with your centromeres.
Mm-hmm.
And that is what we actually see.
Like perfectly, see, it all lines up.
It does that, and that's why it looks like a fusion. Now, you described what you would expect to see at a fusion, and broadly speaking, you described what we do see, and then you said it looks like a fusion only to us, and yet you described it, broadly speaking. So this is, I'm not getting this them and us idea. You were asked what would a fusion look? What would a head-to-head fusion look like?
And Tomkins is claiming it's not a fusion.
Mm-hmm.
And Isaiah, what do you want to describe what you're showing here? And by the way, if Ronnie's watching, those
I'm
are
looking
commas.
I'm looking at the paper he posted.
Ronnie
Not
totally butchered
unlikely.
this when he looked at it.
Unlikely I'm going to be able to understand this and maintain a conversation at the same time.
Hey, everybody. I have something to say real quick. I got to run, but I've never done this before, and it was fun. So,
Armin, thank you very much for joining us, man. We were so very grateful to have you here. We'll have more. So by all means, I do post when I have these. So come join us at any time.
yeah, sure. Thanks a lot. Appreciate it. You guys have fun.
Thank you.
Take care.
Can I say something about the us versus them mentality?
Sure.
Yeah.
I do believe that human beings have bias and prejudice and all the other stuff. You accused me of that in this conversation. Okay? And if you can accuse me of doing that, why should I expect your evolutionists not to have human bias?
Sure, let me address that. Can I address that actually?
Bias
the
prevents
bias is different. The bias is that you've actually said your whole foundation starts from the premise that you already formed a conclusion upon, and you're looking for evidence to support it. If somebody wants to give us evidence against our hypothesis or against our theories, we'll look at it, right? We're not starting from I don't start from the basics, evolution is true. That's a conclusion to me. That's not a premise. You're starting from the conclusion as your premise, and that's not how science is done.
Your premise is methodological naturalism.
Sure, but is all science methodological naturalism? That's science.
No.
You can't just separate it and say, I'm going to form my own type of science called creation science and take out methodological naturalism.
Yes, you can. You
could, but it doesn't do you any good.
That happens all the time in the history of science when people have what Kuhn called paradigm shifts, and they have these massive arguments about the nature of science that are not themselves scientific arguments. They're philosophical arguments about how you do science or what counts as
Can I ask a question?
Yeah.
Question. So if science shouldn't be using methodological naturalism, what should it be using? Methodological supernaturalism.
Science, and I posted this definition, is a pursuit of truth about nature. That is what we should be looking for. We should be pursuing truth about nature. Not
right. How do you start with the
What's
supernaturalism?
nature? What is nature?
If
right.
it is,
we're trying to look at the natural and find natural explanations for what we observe.
But
that's what science
if you're looking at only natural explanations, especially for origins, that might not be the only possibility. And
but that's what science does, though. I think you're missing that. You're not understanding that.
You told me that you agreed with me that science is a pursuit of truth about nature.
No, actually I didn't. Actually, I said that was probably a poor definition. If you wanted to go back and look.
Now someone else said that.
Hey,
and then
can I cut in real fast?
Yeah, go on.
So I just dropped that link. And let's see that this paper is talking about telomere fusion events, and where does it say this? All of the fusion events sequenced involve the deletion of one or both telomeres. The deletion events included the proximal 1 to 2 kb region of the telomere that contains the telomere repeat variants and extended in the telomere adjacent DNA up to the limit of detection. So in other words, when you get a telomere to telomere fusion, you're going to delete the entire telomere and a lot of the subtelomere.
So you lose a lot of base pairs.
Okay, so there's 800 in the human fusion. You're arguing that there's 800 bases of the telomere left.
No, actually, I'm saying that it's of the subtelomeric region, which tends to feature a lot of highly degraded telomeric repeats.
Okay, so you're not saying that saying that the telomere is gone, but it has telomeric repeats in the. So why would that be the fusion site then?
So you understand that the
if it's just subtelomeric DNA that's like a telomere.
You understand that subtelomeric regions include degraded telomeric repeats, right?
You just said that, and I responded to it.
Okay, so if the telomere is gone, the subtelomere with the degraded repeats doesn't work as a telomere anymore, so you can have a fusion event, right?
Okay, so why is the idea that Fiona just illustrated to me with this show that the telomere remained and fused within the telomere?
It didn't
because
remain.
I can't cut the bit of metal off the end of my knife. I did say blunted. Imagine it was blunted.
Well, that's what she said.
The telomere is gone. The subtelomere is A lot of it is gone. Some of it is left. A little bit on either end. That is where the fusion occurred.
Okay, so it's a subtelomeric fusion. The entire telomere is gone. Well, you'll excuse me for being a little confused.
Yeah, so when they refer to it as a telomere to telomere fusion, what they mean is that the ends of the chromosomes merged, as opposed to a big chunk breaking off in the event of the fusion, right? So when you have a fusion event between the acrocentric chromosomes, right? The short arms both break off as the fusion occurs, right? It's not before the fusion occurs they break off and then the fusion happens. It's the fusion itself breaks these two pieces off. But in this case, right, the thing that fuses are the ends of the chromosomes themselves. That's why they refer to it as a telomere to telomere fusion.
I understand this. Where are you going somewhere, or you just think I don't understand?
I thought that's what you asked me to explain. You had asked why, if it's a subtelomere fusion, why do they call it a telomere fusion? I thought that's what your question was.
Well, I think I was referring to Fiona's explanation of it.
Her explanation of it was that the ends, the telomeres, were gone, which allowed the fusion to occur, which is what I'm saying happened as well.
Sure. That's not what I remember.
Hey Ben, let me ask you real quick. Then I want to bring in General Ham Solo and Hogtie here real quick. But are you more consider yourself more a younger creationist or a scientist? That if all the evidence did point this was a fusion, all the evidence pointed that younger creation is wrong, would you give up younger creationism and say, you know what, yes, the science has demonstrated conclusively that this is wrong, or you go in like the more Kirk Wise route that even if all the evidence did point that it was wrong, you would still maintain the younger position out of curiosity?
Part of the evidence is the historical record, and that's not going to change.
No,
That's not the
I
question.
think we're actually
Well, you asked me if all the evidence...
Science. We're talking about the scientific evidence.
And I'm saying that I'm interested in what's true, not just what is defined arbitrarily.
Right, but if the science says that the Earth is not 7,000 years old, that it's billions of years old, would you accept that?
Science doesn't say things like that.
If all
Human beings interpret evidence to mean things like that.
But my question is, if all the evidence you could ever muster by science, everything you could possibly run, every observation we make, every experiment we run...
There's no theory in the history of science that has ever accorded correctly with all the evidence. You're asking
hang on.
a hypothetical question.
Well, there is. There's a consilience of at least 12 different isochron dating methods to get the age of the Earth. And that's not including all the other different types of branches of science. So again, my question is, if all those branches of science do indicate billions of years old with no evidence of young, I'm just asking you, would you be willing to admit that the Earth is not young, or is that your foundation that nothing can change that?
Nothing can change a historical record.
That's not my question. My question is about the
So the
age
answer
of
is
Earth.
the answer is no, because I'm accessing.
Then you can't say that you're doing science. You cannot be objective and say you're doing science if the scientific evidence conflicts what your positions are.
Is Stephen Hawking not a scientist because
Steve,
he refuses to let go of string theory after we found?
If they demonstrate string theory is wrong, he would have to.
They've already pretty much demonstrated that. Look, we're going through.
That's not true.
Because science is complicated in real life. It's not a 1 + 1 = 2 type of deal. It never is.
You said that a historical record can't change. Can it be wrong?
Sure, it could be wrong.
Okay, so you're basing this on a historical record that can't change, admittedly, but it could, in theory, be shown to be wrong.
In theory, I suppose if you... Well, what do you have in mind?
So if you're saying that the historical record that you're referring to says that the Earth is less than 10,000 years old, we could show that that's wrong.
You could have evidence that that's wrong. It's not true that there's no evidence of a young Earth.
Well,
Wait, okay,
Honestly,
so
honestly, I maintain there isn't any evidence, but if you want to bring it to the table, that's fine. But General, somebody wants to ask you a question real quick in the live feed. They want to hear you talk about the historical record real quick, because I mean, we may not have told you, Ben, but like there are Christians. I mean, Fiona's is a Christian. General Han Solo is a Christian.
I don't.
So there's not like this has to do with theology. But General, go ahead and what do you think?
I'm not unaware of that.
Okay.
Jeez.
1 second. Can you repeat that, Steve? Because
Somebody had asked what your opinion on what they mean by historical record and it being accurate as far as the historicity, like the Bible from a scientific point of view. And you're a concordance, so this is a perfect question for you.
Yeah.
What's the question again? Okay. Sorry.
Well, okay, because that question can be fleshed out in many different ways. So are you asking in the context of genealogies, or are you asking in the context of, say, Genesis 1?
Just go with Genesis 1.
Okay, Genesis 1 does not give any dates. It just says it's just a chronology describing from event A to event Z, and I match it with natural history from what science says. It doesn't give. We can date it from Genesis 1. It doesn't give any dates. It just gives an accurate chronology. That's all.
Was there
but
a question?
in regards to genealogy, that's a whole different ballgame. And the genealogies are not base 10 numbers. I can say that much.
I've never heard that one before. That sounds interesting.
Yeah, General's he's into concordanism. He follows the Hugh Ross methodology of looking at the Bible.
Beyond Hugh Ross.
Yeah, who is it? You have some other people too. Yeah,
I've updated Hugh Ross's model without him knowing.
what are they using a different base or just no base at all? Or because I've never had anybody give me a theory about what the numbers in the genealogies mean, and I've asked lots of times.
Yeah, Genesis
other than
5,
what they say they mean.
Yeah, Genesis 5 is a pattern after the Sumerian king list. It's a base 60 number system. Our watch is a base 60 number system, 60 seconds in a minute and all.
Oh, I have heard that before, actually.
When you look at the numbers for the deaths given to each person, there's 10 people mentioned. In the ones place of each value, you have a common value of 0, 2, and 5. That in itself shows that they're base 60. Then you have certain unique numbers given, one to Methuselah, which is a 969 value, which is a triangular number that could have been edited out because Methuselah's name talks about resurrection. From death you get resurrection. The triangular number for 969 is 153, which is the 153 fish that Jesus brings up in John. Enoch's death number is 365. That Enoch, the book of Enoch, talks about how he's a prophet in regards to astronomical signs and so on. Well, that's the solar year, 365 days in a year. So,
but that's not a base 16 measurement, is it?
No, the Enoch value is, but not the.
It sounds like the Bible code stuff I've looked at before.
No, it's not. It's not Bible code.
No, I've actually had a lot of independent people talk about the bases, 60 that they've used. So I don't think it's like the Bible code thing. There probably is something to it.
It's heavily rooted in Sumerian king list numerical patterns. It's not Bible code.
It sounds fairly arbitrary if you're saying that one of them is hex 16 or base 16 and some of them aren't, and just the numbers correspond to everything. Base 60, whatever.
Yeah. It's not hexadecimal.
It's to do with it's to communicate the. So Enoch has a special role. His role is to do with astrological stuff. So he's known as the solar year age. Methuselah's role is to do with resurrection from the dead. That's the 969 value. Lamech is 777. Again, what is 7 in the context of Yahweh?
A hex 60 decimal system? I have no idea. I know what it is in a base 10 system.
There's legitimate scholarship in this. and it's known as Salvation History. Is the title they give this
Legitimate because it agrees with an old Earth.
No,
Yeah,
because of
that's why
ancient
people
Near
do
Eastern
it.
studies.
You know, I noticed you seem to have a lot with this confirmation thing. Like everybody has this. You think everybody has this like mentality that it has to conform to their own
Okay,
beliefs.
I have a reason
and
for this.
I don't know why you would say that because nobody here has given that indication. Is that how any of us are?
I would say that if you want me to answer the question. I would say it because historically Christians nearly always interpreted the book to mean that it was less than 10,000 years old.
No.
Yes, even
that's
Augustine. I know that you're going to quote Augustine and Origen at me. I have quotations that I can pull up for you showing that Origen says it's less than 10,000 years because that's what the sacred writings say, and he had an argument with Celsus. It's in the Against Celsus book. Augustine also argues against an older date of age for the Earth of some 20,000 years by saying Genesis is less than 6,000 years old. Okay? Almost universally, these ancient Christian authors interpreted
Not universe.
almost universally, these ancient
I wouldn't
Christian
even put it halfway.
authors interpreted the book as meaning the Earth is less than 6,000 years old. And from my perspective, it is an arrogant position for a modern person to come along and say, I understand this ancient textbook better than all these people who lived much closer to the actual writing of it than I did. And
Because
that is
it wasn't that way.
modern,
It wasn't
that is
really-
a modern
Just
bias.
quickly though.
A bias
just
of the
quickly.
present against people who lived in the past.
Just quickly, you need to be respective of church father material. Church father material is written by people who evolved over time in their views. So Augustine, I know where you're getting the 6,000-year-old date from. In fact, he didn't make the calculation. Eusebius made the calculation. He's just basing it on Eusebius's chronology. But
So-
later in his life, Augustine does conjecture on the possibility, and I can get you the quote, that he thinks that the 6 days could be 600,000 years.
Yes, I've heard all 600,000.
Yes, he gives the figure that it could be 600, 6,000, 60,000,
Why does he-
600,000.
Why does he say
Actually,
multiples of 6? Actually, could you
Because-
send me the quote? Could you put the quote?
yeah, sure, I'll get you the quote.
It
yeah.
sounds interesting. It's not going to change what I think from one guy, but
All I'm saying is be consistent with the way they used to think, because over time, and it's true today, people evolve in their knowledge and they improve their models.
Well, and
Augustine
one-
was more, he's thinking harder on that later in his life.
if he is looking at just the text, that's fine. But the reason most people, most Christians or believers today deny the historical nature of Genesis is not because they're looking at the text the way Augustine did. They say they take scientific explanations and they say, well, that must be true, and therefore this can't be what the Bible means. And similar to what you're trying to tell me about science is that I'm using information from outside of science to inform my scientific inquiry, which is what creation scientists do. Nobody's denying that.
Mm-hmm.
Because we believe it to be an accurate record, and therefore it should inform what we do in science because it's helpful to us. But like that same way, when you go to a textual criticism format and say, I'm going to criticize this text, you're also taking information from science and putting it into your criticism of a historical text. Science, by the way, which the people who wrote it know nothing about. And it's impossible that they would have known anything about it. It's impossible that could have informed the writing or the interpretation of the text by ancient.
But it isn't unlike flat earthers, right? Flat earthers, and we've engaged with a lot of flat earthers, and they'll say for biblical reasons the Earth is flat, while science has demonstrated that the Earth is not flat.
There's no biblical reason why the Earth is flat.
But would you agree, though, they're saying the same things for the same reasons, regardless if their justification is sound or not. They're saying the Earth is flat. The Bible says it's flat. It's a historical document. It can't be wrong. It can't be changed. It has to be the way it is. Therefore, any scientific data that we find that goes against the flat Earth must be wrong and could be subsequently dismissed. Because that's kind of what I see with the young creationist
But the Bible does not say the Earth is
I'm
flat.
not saying whether it does or doesn't. I'm saying look at the same type of reasoning they use, though. The reasoning is identical. They're using the same reasoning you are. The
It's
Earth is
identical.
flat, and there can be no other way.
It's also identical to scientists who take scientific evidence and insert it into historical textual criticism. It's the same thing.
Well, science is. I mean,
Because
certain things in science we know to be true,
and
right? I
these are
mean, gravity is
philosoph-
gravity, regardless of your textual criticism.
We
right?
know gravity
exists.
We know gravity exists because we can do repetitive experiments showing that things fall. That's what part of my point about historical one-time events that are non-reproducible. Those things do not have the same level of scientific authority that other things do, which you can reproduce. Because that's where science gets its authority. It's because we can see stuff and observe it happening.
Right. We observe stuff to explain things that have happened that when we weren't here to observe them, right? That's what science, like forensic science. Forensic science looks at a crime scene and says, let me recreate what happened. They don't recreate the murder. It's still science.
Right, but it doesn't have the authority of an eyewitness account.
Actually, it has more the authority.
Stop. Eyewitness testimony is the worst type of testimony.
The least reliable. The least reliable.
Absolute worst.
Yeah, by bar none, eyewitness testimony is the least reliable of all types of evidence. I mean, if you have a mathematical proof of something, that is pretty darn good evidence of something. You know, if I say, hey, you know what, the square root of 2 I can demonstrate is irrational, that's a pretty good evidence if I can come up with a proof for that, which isn't.
There's no such thing as a mathematical proof in science.
Well, there are mathematical proofs. There's no scientific proof, but there is mathematical proof.
There's mathematical proofs, but there are no such thing as scientific proofs. You would agree with that?
Yes. Science doesn't prove anything. It only explains.
So there are levels of uncertainty in scientific knowledge as well.
Certainly, there's going to be
it's a matter of opinion. It's a matter of choice. It's a matter of belief. Dare I say it?
Let me ask you this.
Which types of evidence you choose to accept as more valid than the other?
Well, I do want to ask.
And I choose to accept historical evidence as being more valuable and more pertinent when we are talking about historical events.
Let me ask you this real quick.
I think that is rational and it makes sense.
Do you agree that we'll say there's a dozen or so isochron methods of dating? Do you think that the dating methods are sound? Do you think that they actually indicate? Let me actually rephrase that. Do you think the dating methods actually indicate an old Earth?
No.
Okay. Why is it that the rate study, which is a young Earth creationist based study, the rate study, radioisotopes of the age of the Earth, they even admit that the isochron dating method does work?
Yeah. Well, the isochron dating is an attempt to pretend that, it's an attempt to make it more rigorous so that you're not depending as much on assumptions, but the assumptions are still there.
There's still assumptions in that kind of dating.
There's assumption, and it just goes to show that there are assumptions in science all the time.
But the dating method works.
You look at me and you tell me that I am believing this and that's not scientific and blah blah blah. To me, that's just semantic.
But the dating methods. My question was, the dating methods indicate an old Earth, and even creationists admit that the dating method indicates an old Earth. Yes, as assumption. You have to know the parent-daughter ratio of the radiometric isotopes and the non-radiometric isotopes, and plus there's like 2 or 3 different things you have to assume. But the method itself is sound, and that the method itself does indicate an old Earth. Even creationists admit this.
It's evidence, yes.
Okay.
It's not the only evidence.
But even
it's not the only evidence, because you referenced the rate study.
But even they're saying that the method works. This is all saying that, yes, there's evidence out there that there's an old Earth, and they're trying to explain it away by other means, which we can get into, which I don't want to, but they're trying to explain it away by other means. But at least they're admitting that the evidence shows an old Earth. Do you disagree with that?
Particular line of evidence.
Yes, that particular line of evidence.
There are other lines of evidence, such as the rate of helium diffusion.
Go ahead, Fiona, and then we can get into helium diffusion too, if you want.
It's something that was said there, and I can't let it pass.
Go ahead.
You talked about radiometric dating methods, and you said that it was a way to pretend something. And I have to ask, what do you mean by pretend?
It was a way to try and wash out, in other words, to make the basic assumptions of those dating methods more supported, I guess I would say.
Right. So
I mean, is
so,
that accurate? That sound. I mean, I don't think anyone would disagree with that.
So knowingly, scientists pretended. And this is what.
Well, okay.
I'm
They're
not
trying.
getting.
Maybe I used language that was unflattering, but it's attempting to make their conclusions more solid.
Well, the conclusions are solid
Why?
from the dating methods. I mean,
And
again,
the
you have to
assumptions that
invalid-
you use.
Right, but the assumptions, there's three, and I can't remember all three off the top of my head. I haven't looked in this a while, but there's two that I do know for a fact. One, you have to assume a constant decay rate, and you have to assume that the ratio of parent to daughter from the radiometric isotopes that are radioactive to the non-radioactive ones are constant. The ratio is constant, and those are pretty fair assumptions. Now, what the rates study did was try to invalidate the assumption that the decay rates have changed. And that opens up a whole different can of worms. That is a really strange way to try to invalidate radiometric dating, because if they were right, you have to account for an awful lot of energy over from deep time to 7,000 years, and that amount of energy would be tremendous. It would be enough to pretty much liquefy the
But
planet.
they're doing so because of the evidence. Okay.
But
Yes, it presents massive theoretical problems, but we also know that physics
I
has
wouldn't call
lots of
it theoretical. The math can actually demonstrate this.
It's based on physical theories that use the math, and we know that. I mean, the only reason physicists have been chasing string theory down a deep dark hole for 40 years is because
I'm not a fan of string theory, so that has nothing to do with what we're talking
about.
Don't
work. It does.
String theory is not a theory. It's only hypothesis at this stage.
Because physics is not complete.
Nobody's saying that's complete, but the radiometric we understand. I mean, we know it works because atomic theory works. I qualified reactor engineer on 2 different reactor plants. I was supervisor for D2G and S1C. I know from experience
and
that
I know that's why you
fission
brought
works.
this up, is because this is your big issue.
That's what I'm saying. We know it works, and when you have this thing that people are saying, well, the decay rates have changed. Okay, you know what? If that is, demonstrate it, right? And that's the issue that I see, is I don't see them demonstrating anything that they're saying, oh, the assumptions are wrong. Okay, well, why are the assumptions wrong? And if they were wrong, what would be the ramifications?
Because the rate of helium diffusion is too slow.
No, we can go into Humphrey's work as well. That was he used the wrong geometric formulas. He had 5 major mistakes.
He responded to all of that. I got into this on the Creationism forum a while ago. He has responses up to all of that. And part of the problem with engaging in a point of view that you don't agree with is you have to sort of live inside the opposing view's assumptions, okay? And so doing that, there's going to be multiple different scenarios that come out of that. And he had a whole response to somebody who actually submitted a response to the Journal of Creation saying, here's your problem with, the Q-OT ratio or whatever, and he posted a response and all of this. That's what normal scientific journals do, and that's part of science.
I know more about fluorine halos than I do.
And this is all great, but the thing that I react against and why you sense negativity from me is because we are participating in scientific discussion, and you refuse to allow that we are also doing science. And you try to make that argument.
But Ben, you just admitted though, Ben, you've admitted earlier though, if all the science that you claim to love indicated that you were wrong, you wouldn't accept it. How can you claim to be scientific then?
I wouldn't change my mind about what I think is true. That doesn't mean I'm not doing science.
That's exactly what it means.
Because you think that science.
Conversation on that point. You already now have a bias that.
That's exactly true.
Everyone has bias. You think that science is the ultimate epistemology. That scientific truth supersedes all other truth.
No, that's not. See, that's when people make assertions that are not true. There's nobody here that follows scientism, I can assure you.
But
Okay,
you
so that's
asked
not our epistemological framework.
you asked me if all the scientific evidence says the Earth is old, would I change my mind? And then if I said I didn't,
Then
you
you're not being
say
scientific. Science, you conform. If you're doing science, you have to conform
no,
to what
no, no,
science
no,
is
no,
telling
no.
you.
You're asking me that I have to believe something in order to do science. That's not true. You can do science.
How many,
You're
what
introducing
is,
a dogmatic that I'm a heretic, okay,
by your
and that I
logic,
can't
you just
participate in scientific discussions.
No, that's
because
not what I'm saying at all.
I have beliefs that are not based on scientific evidence.
Yes,
I-
Ben,
and that
by
means
your
that
logic, you just refuted the 2 or 3 witnesses rule in the scripture.
What?
You've just totally neglected and thrown out the 2 or 3 witness rule in scripture by your logic.
Is this supposed to be automatically clear or? Drop.
Yeah, what is the 2 or 3
Elucidate real
witness rule?
quick.
You can make a conclusion as long as you have multiple lines of evidence.
Were there multiple biblical authors?
What's the rule state in the Mosaic Law?
I know the rule. What are there multiple authors of Genesis?
Okay, so for
Answer the
example,
question.
What?
Are there multiple
talking
authors
about-
of Genesis? Do you think that
Quite
the author of
possibly.
Genesis? Well, do you think that the authors of Genesis weren't consulting multiple independent lines of evidence? I think they were.
And what are
And
you
this
trying
is
to make
part-
a point from that? I'm trying to connect it to
You're asking-
scientific empirical evidence here.
No, you're asking me about the 2 or 3 witnesses thing.
Yeah, what is the principle? The underlying principle is to say you cannot make conclusions unless you have at least 2 or 3 witnesses or lines of evidence. So, when Steve and Fiona and everyone in this room give multiple lines of evidence from various schools of thought that the Earth is indeed 4.5 billion years old, are you just going to ignore those lines of evidences and ironically hence ignore the scripture that demands that you take into account those 2 or 3 lines of evidences?
So you guys are the only opinions that count, or there are no other people who have other lines of
Yes,
evidence that show a
all
young
the
Earth?
disciplines. So biology, geology, physics, astronomy.
So I
And
should
that's at
decide.
least 4 witnesses already.
I should decide what is true based on a popular vote, or a popular vote among people
This
who
is
are
not a
considered
popular
experts according
What
to some
is
criteria.
Is evidence a popular vote?
You,
Hang
you're
on.
just-
Hang on. Fiona's taking off, so I wanted to thank Fiona for dropping by. Fiona, thank you.
Yeah, I'm going to wander off now.
I know it's late for you. It's like 3
Yeah,
o'clock
it's,
in the
it's
morning.
very
for you.
late.
Yeah.
2:30 in the morning. But I'd just like to say that I enjoyed the discussion about the chromosome fusion. But I do think that, I mean, we all need to remain open-minded and be willing to look at all the evidence. Science is tentative. Science doesn't declare truth. It's always tentative based on the currently available evidence, and that can change. And unless you accept that tentativity, then you're not doing science.
I
And
would
with
agree
that,
with
I
that.
shall bid you good
All right.
night.
Thank you, Fiona. Good night, Fiona.
Take care.
Hey, Hogtie, you back? Yes, I was going to check with you.
That's part of my point she was making for me.
Let me see if Hogtie's back real
You
quick.
guys are so concerned that I believe something is true.
No. I think
Because
you're
that's what the scientific evidence shows, or you
I
think
think
it does.
I think we were misunderstanding what at least I'm trying to say. Science.
You're asking me if I would change my mind.
Right. Well, because if science is indicating something, right? I mean, the people back when they said that the sun revolved around the Earth, they were adamant about that as well. They were like, you know what? Yeah, of course the Earth cannot revolve around the sun. Therefore, any evidence we find has to, support a geocentric type model. And then, of course, hundreds of years later, we're like, oh, yeah, of course they were wrong back then. That's how things change. And I think that 500 years from now, nobody's going to ever be thinking about young Earth creationism. It's just not going to exist. I mean, you have to look at least and say, okay, there's all this evidence, which I've even said the dating does indicate an old Earth. Now you could try to falsify that, which they've tried to do. They've not succeeded by any means. In fact, I think they've actually done the opposite. But
I would admit that is one scientific problem with young Earth creationism, and another big problem is the starlight issue.
Starlight's a huge issue.
As far as I'm concerned, though, I believe those are really the only two major issues with young Earth creationism, and if that's all that's wrong with it, we're doing pretty well.
There's a lot more than that. But
of course
let me ask you this:
obviously, shows that the radiometric decays don't change.
Yeah, see, the problem with I don't know if you know this, but the problem with the radiometric decay rate changing is if it changes the fine structure constant, which changes the speed of light, which changes all the Planck's constants, all the natural units. It'll change the Boltzmann constant, it'll change the gravitational constant, it'll change everything that we basically hold as a constant. It'll have to affect all those. Mm-hmm. And the problem with that is if you have that kind of things that affect the electromagnetic constants, then you can have things like fusion. The sun will not hold up because you cannot even overcome a Coulomb barrier. So we know that these things have not changed, because if they did change, it would have massive repercussions everywhere. And so when people are using the fine-tune argument to say, oh, look, the universe is finely tuned for us, and in the corner of their mouth they're saying, oh, look, but decay rates have changed, you can't have it both ways, because we exist. And if we exist, the decay rates could not have changed the way they're suggesting. They just could not have. It would have been a violation of everything we know about physics.
Okay.
Yeah,
they're suggesting that most of this happened during the creation week, is my understanding of it, which is not dissimilar from a Big Bang theorist suggesting an inflation field which doesn't exist anymore.
That happened in 10 to the 36 seconds or something.
Right, and it would have happened initially before all that stuff you're saying has massive consequences would matter. It would happen in probably the first creation day, when he's creating light or creating matter, maybe. That's what that means. Who knows? But it wouldn't have the consequence. The sun was...
physics?
I'm saying that a creation of a matter would have to violate the laws of physics, so of course there were strange things happening during creation.
Well, yeah, but if we look at...
There are strange things happening during the Big Bang.
But when we look at the universe, and I want to get hog-tied here real quick. But when we look at Hubble's constant, which is like 70 megaparsecs per kilometer or something like that, kiloparsec. If you start messing with these things, right? If you want that variable speed model to work, the one that Hovind uses.
I don't hold to a variable speed of light model, so I don't know what relevance this has to anything.
Well, because a lot of young earthers do. They actually promote this, and the fact that you would have to change you would have to change Hubble's constant to 160 million from 70. So 70 to 160 million is a huge change.
I saw your post on that in the group, and I posted a video from John Lyle. Did you watch that video?
No, I don't remember you responding.
Okay.
But what about it?
I'm
not big into watching videos, because if people can't really explain the problem, I'm not going to watch a 5-hour video on something.
It's like a 45-minute presentation on the different possible solutions.
Name? John who?
John.
Who?
John Lyle.
Oh, I thought you meant Hartnett, because usually Hartnett's the second person to do it.
I don't know how much longer do you have out of the way? I usually don't keep these things more than 3 or 4 hours. I know we've gone on for a while, and I don't mind. I mean, I could be as long as you want, but I don't want to like overtax you. So are you hanging in there? Or did we lose his mic? Ben? If Ben's talking, you can't hear you, Ben. You might have to reload. Hit F5 if you can hear me, because we can't hear you.
Oh, Jason Lyle, you mean, not John Lyle.
Jason Lyle. I'm sorry, you're right, yeah.
Yeah, Jason Lyle, yeah. Jason Lyle and John Hartnett, they're models, yeah.
But I think we may have lost Ben's mic.
Or like you're saying about the time, maybe he just fell over. I think sometimes you get someone in a room like this for, where they're kind of...
Yeah, he's not used to it like we are, right?
Obad has a really good question. Would the change of radioactive decay rates change pi?
According to Ronnie, probably.
So what did it mean to what did that mean to the ancient Hebrews? If you don't know, you're wrong.
Seriously,
By the way,
I saw that.
I have to throw this announcement out there, as we all know. And well, you know what? I'm not going to bring it up right now. I'm going to bring it up next hangout. I don't want to bring it up this hangout.
Wait. Would the changing speed of light affect the rate of DNA degradation in water, though? I mean, is that possible?
With Ronnie, anything's possible. But I don't know. Are we ending this? Did Ben lose connection? Because I think he kind of dropped mid-sentence, and I think he might have a connection error. Did you get a chance to listen to much of this, Hawk?
I didn't. I've listened to about the last 20 minutes of it, and I do kind of feel like I missed a good discussion on the chromosome 2 thing.
It was good. It was really good.
The part that made my ears perk up was his mention of bias. Ben's mentioned it a couple of times, and I wanted to ask him a couple of things about that. I mean, I accept that I'm coming in rather late, so if he's kind of had his fill, then I would understand without accusing him of being a coward, as the folks on the other side would tend to do. Hopefully we'll cross paths on another occasion when I can ask him about the bias thing.
He came across as quite arrogant, to be honest.
No, I
And
think he did
didn't
okay.
listen to it.
I don't think he made his arguments at all, but I don't think he came across as arrogant. I mean, all young creationists are in some degree arrogant because they seem to be indoctrinated that their belief supersedes reality, and that's a scary position.
You've got the truth in your pocket. All these other people are fumbling around, with you sort of picture everybody else. If science is a flashlight, they've all got flashlights with, weak batteries and dim bulbs, and they're sort of fumbling about trying to find the truth that you
Hey,
have
Hogtie,
access to.
don't call these people a bunch of dim bulbs. That's not nice.
I said, we're the dim bulbs.
Well, I do think they have it. I'm not accusing Ben of this by any means, but I've seen this a lot with young Earthers, that they seem to have this impression that they know something everybody else doesn't know. They feel a little bit special. That look, all of these 99.7 or 8% of all scientists, they don't know what they're talking about, and they feel like it is part of an elite group of people that know the truth. And nothing's going to sway them from that truth. Nothing. And he even said that even if all the scientific evidence went against him in the creationism, he would be still, into young Earth because that's his foundation. I don't know how you can live your life that way, though.
I think first off, how do you live your life knowing that you have access to the creator of the universe? I mean, that's
pretty impressive, actually, if I think about it, Hawkeye.
Yeah, but then of course dealing with the other people around that just don't seem to get it. I thought you were on the right track, like when he mentioned bias, and again I sort of perked up. I thought maybe I will join this hangout because I wanted. It's an issue I like to address with them, and but I think you were on the right track, Steve, when you were mentioning about that. Like, I mean, yeah, people have their sort of current understanding, and that may color their observation of new facts as they arise. But things are drawn from evidence toward conclusion. And I think the point you raised, Steve, about starting from a conclusion and then saying, well, now how do we look at the evidence? How do we interpret this evidence? Oh, and then you find some evidence that you just simply have to just dismiss, just ignore, pretend it's not really the case. And I mean, I find it strange because they will actually publish. And one of the things I had on my screen as I was joining was the principles of scientific creationism from ICR, from the Institute for Creation Research, and it just says, each of the major kinds of plants and animals was created functionally complete from the beginning and did not evolve to some other kind of organism. The first human beings did not evolve from an animal ancestry. When you have these things, it's like, well, no, these are foundational. In my view, that's the thing that. So we've got Ben back in here. I think the point's made. You guys know what I'm getting at. I think you're on the right track with that, Steve. Is saying
thank you.
That's really the critical difference between sort of science and not science. But I think we had a couple of questions there for Ben before I proceed with that.
Oh, I don't remember where you left off. I think Ben, you lost connection. It looked like when you dropped. I was asking you how much kind of time you have left if you want to try to suss anything else out. You might have to unmute yourself though. There you go.
I'm good.
Yeah.
Okay.
Yeah. Don't worry. Google. Google's sucks. Let's just put it that way. Google Plus, these Hangouts, they're very touchy. But
No, it wasn't Google. It was a issue with my computer. It it's not kind of embarrassing. I actually knocked the power cord out.
oh, we've all done that.
So
Yeah, I'm on a bad connection here as far as my electrical thing. If I kick the wrong way, it'll actually go off.
yeah, I put my computer up on a chair and I put my feet up on the chair a lot. So
I'm going to be asking questions. Let me ask you first. Again, do you have like another like half hour or so? I mean, it depends on your time thing. I don't want to overload
Sure.
you. Okay, cool. Because I mean, we do it for
I
long
love
haul. You have
doing
no. I don't like having long special events because people do watch these afterwards, and I don't want somebody to sit down for like a 5-hour hangout. But since I don't want to start another hangout, I'm going to continue this one on. But I'm going to ask the audience that's listening. We have about 40 people. If you guys have any questions, but before they start answering their questions, I wanted you to give your best example of what you do think that is positive, and let me reemphasize this: positive evidence for a young Earth, not negative evidence against an old Earth.
that's a
I'm
good
curious.
question. I'm looking at a couple extra. I've already made the post about the helium diffusion rates. So if you want me to explain that, the
Sure.
helium diffusion rates. Well, helium is a alpha particle, correct? But without the
No,
electrons.
well, yeah, without the electrons, right?
So-
helium has a neutral state, so it's alpha particles, a helium nuclei without the electrons, right?
Yeah. So it would rapidly acquire the electrons once it's done decaying. So it becomes a helium atom after the alpha decay occurs. So it's a decay product. It's a product of radiometric decay. And what they looked at, the rate study looked at, was levels of helium in zircon crystals, which is, you use zircon crystals for dating. And they found that these levels were relatively high compared to what you would expect based on helium diffusion rates. And so they looked into helium diffusion rates and all of this.
This was Dr. Humphreys, by the way, right?
Well, it was several of them. Yeah, they got $1 million for this study, which is a massive amount of funding for ICR. That's why I laugh at you guys when you say they have so much money. They really don't.
$1 million for a helium diffusion study? That's quite a bit.
Yeah, well,
Which
they sent their samples to I believe it was some geochemical company in Russia, because he was the foremost expert on helium diffusion or something. I can't remember, but so the idea is that if helium diffusion is occurring at a constant rate, which is an assumption, then over billions of years, most of all of this helium should have diffused out of the rock if it was a decay product from radioactive isotopes inside the rock, and it didn't. And they determined that all of the radiometric, this is why you say that they accept the accuracy of the method, is because in this conclusion they accept that all of the radioactive decay occurred because the helium is there, but the helium is there and it shouldn't be.
Right, but the diffusion method, I mean, this gets into an area that I haven't looked at that much, so I'm not going to talk too much on it. I remember reading up on Humphrey's work. I know that the formula they used was a wrong formula because it had a different amount of surface area for the diffusion rates. And as you know, the more surface area you have, the higher degree of diffusion. That was one of the bigger problems. But the biggest issue that Humphreys had with the studies is that they were trying to account for the energy from deep time. Same what you would have with any decay constant problem. They're trying to invalidate the claim that decay constants are pretty well constant for the most part. There are some things that can change them, K-capture, and it's nominal. It's like 0.2%. But they're trying to say that the decay rates have changed, and they're trying to account for all this energy. And you know what? They haven't been able to do it yet, Ben.
That's correct.
And
that's
so
a theoretical problem.
That's exactly it.
The evidence shows that the decay occurred quickly. Theory is supposed to match evidence.
See, I don't know enough about the helium one. I know if you want to talk about polonium halos and use that argument, then I can discuss it ad nauseum.
I didn't look into that, but I'm familiar with the radio halo argument.
Because the radio halo argument is easily debunked. I can easily debunk that one.
Well, shoot.
Okay.
I'd be interested.
What What Gentry is trying to say is that there's different isotopes in the decay chain of uranium-238, specifically polonium-214, 210, and 218. That he is finding these things in biotite, type of granite, and he's saying, okay, these halos exist without a primordial halo from the parent elements, and sometimes they do. Instead of having a big halo, which would be like the uranium, which has a very intense mega electron volt of the alpha particle, where you have this big old halo, 360, halo around the part of the which has a nucleation point. If you do a cross section, it's going to produce a circle, which is going to be that halo, right? So when you see his photos, he's actually just taking a cross section of a three-dimensional 360-degree sphere. Well, his problem was he didn't recognize that the polonium-210 and the radion-222 halos were identical as far as their energy levels. They are pretty much indistinguishable. They overlap. And he only had a 2-micron ion probe to even try to do the resolution for it. So he negated the fact there was even radion-222 in these isotopes. And the problem with that was, when you find these polonium halos, even when you find polonium-210 by itself, they're always going to be on fracture sites, microfracture sites on the granite, which means something has escaped and diffused through the rock and then degraded or disintegrated later on. So the radon-222, which is a gas, can actually go through microfractures and actually be dissolved in water, and water can disseminate it through at the rock as well. And so he didn't take that into account, that there was a mechanism by which these polonium halos could be produced without having instant solidification in a primordial 7,000-year-old hypothesis that he had. And when they brought this as attention, they said, "Okay, look at, this explains what you're seeing. This explains the fact that you couldn't reproduce the perchlorate discolorization. He didn't have the right energy levels of the alpha particles to do so. He tried to kind of ad hoc it. He never was able to actually reproduce these things exactly. And the reasons being was he didn't take into account what's called the Briggs effect. And the Briggs effect is basically what you were saying. You know how you're saying that the alpha particle over time is going to start getting absorption of the electrons and it's going to become neutral and become helium, right? Well, until it does that, it's a very ionization particle. It produces a lot of ion pairs because it's highly charged. It's got a +2 charge to it. So it's going to ionize a lot. And then as it toward the end of it, it's going to start getting deionized because it's going to absorb an electron, and then it's going to start breaking. The speed's going to break. So it's like a breaking radiation. And then it's going to have a little bit on the end of that's going to cause a little bit more damage. And so there should be a very set discoloration pattern. And he just never could produce this. His explanation for all this, when it was brought to his attention that there are other explanations of natural explanations rather than invoking that it happened instantaneously, spontaneous creation, he invoked what's called singularities. Have you heard of those?
In physics as a black hole, but
no. There's a singularity is a one-time creation event, and so basically he said, well, God did this. He did it as a one-time creation event. It's called a singularity, and when he did that, you agree, that's no longer in science anymore. That's no longer following, whether you agree with it or not. Science follows methodological naturalism. It took it out of that form. He actually at that point falsified his own argumentation, his own findings, because you know what? He said the way we used to say this is the science, this is what physics is showing, and now he's saying nope, the physics doesn't show this, but God did it this way anyways. That's not how you do science. You cannot use God. You cannot posit a deity to be an explanation for your observations when there's scientifically naturalistic explanations to be had anyways, right? I mean, we could explain these halos. We'd explain exactly how they form and how radon-222 is the mechanism by which it leaves the halo from the uranium-238, goes through these microfractures, and then starts disintegrating into 214, 210, or actually it's 210, 214, and 218. So, he doesn't even use these things as an example anymore. He can't say that that physics shows that there was a spontaneous formation of the Earth. He can't say that anymore because he doesn't have science for that. He hasn't used that argument in a long time. He hasn't been around in a very long time. These things, these were brought up in the 90s. I remember discussing these in the early 90s.
So it's evidence that we're not being scientific if we accept criticism and stop using an argument that may be flawed.
He doesn't use it anymore. I haven't heard a peep from him. He's even still alive. He basically, like I said, he took it out of the realm of science and said, okay, well, yeah, okay, there's an explanation of this that makes sense naturally, and he didn't have the right tools to even find out whether this was right. It wasn't until later on they did more studies and realized that there was a naturalistic explanation.
It's not proof that creation scientists are not being scientific because we
But they're
may
still
have
using
done
this as an
something
example.
wrong.
People are still building up polonium
People
halos.
are. You just said he wasn't.
Right, but
Random
but I still hear
people
but
on the internet.
But this is one of those things that was used as a young Earth creation argument for a very long time. It has been very, very debunked, thoroughly, utterly debunked.
Well, I'll take that under advisement from the source.
Well, I mean, you can go read his book. Creationist Tiny Mysteries. He even mentions that.
I'm talking about the debunking.
He
consider
debunks
you
himself
to
in
be
his
a
writings.
biased person.
In his writings, he admits it's supernatural in origin. It automatically now takes it out of science. There's no debunking to be done. It's not science at the moment.
When he says that, is he talking about the first couple days of creation when it maybe have occurred? Because we already posit that there are all kinds of supernatural events that occurred in the creation week.
Right, but you can't test those as science, and there is explanations. The
lots
polonium
of-
only lasts about 3 minutes. Okay, they're not very long half-lives.
So it would have occurred in the first creation week.
It would have been, yeah. That's why he's saying these halos had to occur instantaneously to be frozen in situ like that.
Let me meet you halfway here. If what Gentry is saying is that God arbitrarily formed polonium halos for no reason, then I don't agree that is creation science.
No, I don't think that's what he's positing.
In the Bible, when God does miraculous things, it is always for a practical, immediate reason. Okay, and this is part of the C.S. Lewis's argument. C.S. Lewis's argument in the book Miracles, which you should check out.
What I would find odd would be if and again, I don't find too many young Earth creationists using this as evidence anymore, but there are a few. But what I find would be odd is that why would God use polonium halos as evidence for young Earth, thinking that people are going to be understanding things about polonium halos? It's not an easy topic. And that's one of the things to me is like, what if there's going to be evidence for a young Earth? Shouldn't there be evidence for a young Earth that's kind of like open to a lot of people, not something you have to have some kind of a deeper understanding of? Like, when it comes to the fusion site, you kind of have to know a little bit about biology about it. The average person is not going to understand what we're talking about when we're talking about telomeric to telomere fusions. Correct? Would you agree with that?
I do not believe. Well, yeah, but I do not believe that God put stuff intentionally into nature to prove that he exists, except in historically documented cases where they were readily apparent to ancient people, which is going along with what you're saying. Okay? I do not believe that God, for instance, created light in transition, showing events that never really occurred. That's an argument you hear from random church-going people who believe in a young Earth, that God created light in transit.
Right.
And if you watch John Lyle's video, he states the problems with believing that. So we're not insensitive to that sort of thing. It's not some kind of hand-waving, all creationists can't participate in science because some people invoke miraculous explanations whenever they want to, arbitrarily ad hoc. Okay?
Well, nobody's saying creationists can't do science, but to be a scientist, you have to conform to certain basic things that you're trying to find a naturalistic explanation. That is what science is.
What I'm getting from you is that in order to be a scientist, you have to believe that science, the evidence, shows you things that are true, and you must believe it, or you're not doing science.
No, matter of fact, the whole point of being science is that you have to refute your own hypothesis.
Then why did you ask me, would I change my mind about the age of the Earth?
Because if the hypothesis has been invalidated, you form a new hypothesis.
But there's extra scientific
and
evidence.
Creationism has been falsified to no end. The hypothesis is no longer valid. You mean a scientist, when given enough information, is going to change their hypothesis? Because you don't prove a hypothesis. No hypothesis ever gets proven. What you do is you have to be able to reject the null hypothesis, and by doing so, you accept the claim of the hypothesis. Right? It doesn't prove it. Right? The hypothesis isn't proven. It's just confirmed such that we accept the model that it works. Okay. But if something came later on and falsified that, right? If something was completely a paradigm shift. Like, for example, catastrophism vice uniformism. That was a paradigm shift back in the 60s.
Distinction now, but
well, it's not. No, it's not, because what
it
a
is
person,
because
when
well,
a person,
this is a rabbit trail, but it is because, for instance, when evolutionists want to explain the extinction of the dinosaurs, they posit a catastrophe, catastrophic.
But that's not what
event.
catastrophism is, though.
Yes, it is. It's when a meteor strike hits the
Right,
Earth and
but
throws up a bunch
catastrophism
of dust that blots out the sun and all the dinosaurs.
Right, but that's
are killed.
that's not from
That's a catastrophic event.
It's a catastrophic event, but it's not. But catastrophism was the position that these things form over very quick periods of rapid times, like boom, something happens, and then there's a time nothing's going on,
like
and then
a
the
meteor
next
crater,
time. Yeah, but that's not. But that does happen, right? Plate tectonics explains the Himalayas, right?
That's what I'm saying.
But we know that hang on. We know the mountain range didn't occur because of catastrophes. We know that some things can form catastrophes. We do have catastrophes, but that's not what catastrophism is. There's a lot of catastrophes out there, right? You mentioned one earlier, Pinatubo.
I don't think there is such a thing really as catastrophism. That's a word that older creationists use because they're still arguing with Charles Lyell or something.
Well, that was actually the science people used those too back in the 60s.
Well, I think
plate tectonics.
I think it's honestly, I wish people would stop using those terms because it's not really that helpful. It's the idea that there's some kind of, it's similar to methodological naturalism. The idea that there's some kind of arbitrary system that we have to use in order to correctly do science. You're looking for truth. There are lots of ways to use logic and evidence to get there.
But here's again the
if
thing
you
you're
have
not
a
understanding.
catastrophic event that explains something.
There's different ways of going to truth. Science is one method. Science adheres to methodological naturalism. If you don't want to use methodological naturalism, that's fine. You're not doing science. You can use some other method. You can use philosophy or something, right?
I don't agree with that.
But that's what science is. That's
what you say science is, but can you scientifically prove that? No, that's a philosophical position.
Well, because science don't prove things. But do you think science should go into the business of having supernatural causation? Oh, we don't know how to do this.
If the evidence suggests that.
Has there ever been evidence for supernatural causation in the history of man that we know of? Like what?
Time.
Like what?
Like the confluence of physical constants that are required to be finely tuned.
How is that supernatural causation?
Because it's a correspondence between a very low probability of something and a very high specificity of an outcome.
Probabilities
corresponding to the possible values of these constants.
I would say the probability is 1.
Well, if that's the case, then someone would have figured out why it's 1, and that would be called a natural law. But that's not the case.
Just because we don't know something why yet, that's an argument from the God of the Gaps, right? I mean, that we don't know something, that doesn't mean it doesn't have a natural explanation. Science is dedicated to finding naturalistic explanations. That's what science does.
But it cannot be naturalistic explanations all the way down, especially if the Big Bang is true, because that's an event when everything started. And even Hawking and those guys who come up with M theory and say, oh, well, we have this theory that says because of the law of gravity, something can come from nothing. Well,
that's not that's not what they're saying. First of all,
that's an exact quote.
No. First of all,
it says because there is such a thing as gravity, something comes from nothing.
No, listen. First of all,
and it's his fault if he's not being precise.
We don't know there's anything called nothing. We've never experimented with nothing. We've never discovered nothing. We don't know what nothing would even entail. We have no idea the concept of what nothing would be because we don't know if nothing can exist. Okay, when people like Krauss and the rest of them talk about nothing, they're actually talking about a quantum level state, right? A level of quantum fluctuations. The Big Bang doesn't tell us.
That.
Contrary to what people read on Wikipedia and other things, the Big Bang does not tell us the actual origin of the universe. They use that term very colloquially and very loosely because it explains the period after the universe is here, the inflationary period. Okay, and there's a lot of different periods: the reionization period, the recombination period,
requires an inflation field.
the
That is
hybridization
arbitrary.
period. Yeah, but the model, right? The model itself predicts certain things, right? Just like all models, and it predicted the cosmic microwave background radiation at
And that makes it
2.73 Kelvin.
That makes it a scientific theory, regardless of whether or not the prediction is confirmed.
But it was it was a
doesn't-
confirmed scientific theory
it
after
doesn't-
they discovered that, correct?
It doesn't make it non-scientific if a prediction is not confirmed.
I
Would
didn't
you
say
agree
it
with
did.
that?
Yeah, I did. Yeah, I didn't say it
So
did. But these
science-
were that was confirmed though, right?
I'm not arguing about all that. I'm not a physicist. I'm not even close to understanding that sort of thing. I do believe that I would have to become one to decide whether or not I agreed with you.
Well, does anybody else want to?
I'm going to be
chime in on that?
agnostic on that.
I mean, you have heard of the CMB, right? The cosmic microwave background.
Yes.
Okay. Do you know why that's evidence for a Big Bang? You know why they say it's the confirmation?
I don't know why particularly.
Okay, because during one of the periods of the Big Bang, very very early on, the universe was in this opaque condition. Okay, because there's electrons forming everywhere, but there was no way for these photons to actually get out of this little opaque area. Okay, they were confined there.
Because of gravity?
No, because of it was just I don't remember the technicality specificity. This is part of the recombination period, but after the electrons started binding with protons and forming hydrogen. The photons were allowed to escape, and they did so automatically in one big burst. Okay, and so the opaqueness of the universe stopped. I got a dog barking. Sorry. And all these photons were released. Well, they had hypothesized if that was true, we would see this ubiquitous background glow, a microwave background radiation around 2.73 Kelvin. Guess what they found? A ubiquitous microwave background radiation around 2.73 Kelvin. The prediction was spot on. And if it wasn't something they could predict by the model, then yes, the model would have been confirmed. It would have been a possible model. But this was a model that made a huge novel prediction.
Mm-hmm. That sounds interesting. The theory of
reionization.
that,
I think you're
that
right.
I think
the reionization period, not the recombination period. The reionization period.
What I actually think is about the starlight issue is I think that we're looking at gravitational time dilation because of a massive expansion of the universe, which is similar to a Big Bang. Now, I don't know all the details, but because of time dilation that I do know about from relativity, is that things which approach the speed of light slow down, that it's possible that
well, how would you have time dilation when it's actually the inflation of space-time itself, right? Time dilation does exist. Okay, there's Lorentz contraction, but you get about 20% of the speed of light, you start having time dilation factors. Okay, but the universe itself was expanding faster than C at the time. Okay, that was space-time itself.
Nothing can go faster.
Nothing with mass.
Okay.
Yeah, nothing with mass. This Einstein actually did allow for the fact that space-time could go faster than the speed of light. Matter of fact, there's actually a guy named Acuberry right now. He's a Mexican physicist who's developing a warp bubble on that premise.
That's some things that people from your side often say, is that nothing can go faster than the speed of light, and relativity says this, but that's not
nothing with nothing with mass or information go faster than the speed of light. Right? I mean, a tachyon is faster than the speed of light, and it's a hypothetical particle that they've had hypothesized for decades. Do I necessarily believe it exists? No, probably not. But I mean, you can't say that.
It was in Star Trek.
But you can't say that. Oh, your side says this. No, because a tachyon has been hypothesized for decades, and that goes faster than the speed of light. So things can go faster than the speed of light, but nothing with mass can break that barrier. If it's on the other side of the asotopic line, it can exist.
Random people on the internet who are arguing against creation.
Well, then you need to set them straight, right?
Then you set your guys straight.
I can't fix everybody, man. We only have so much reach to people.
Look, the cultural issue that we're dealing with here is that there's a large segment of the population, 40%, who are young Earth creationists, and that's in the United States, and it's been at that number since 1980, and of course before then it was larger. And
that's part of the Pew poll, but I think that number's a little high. Honestly.
These people are constantly berated and made fun of and called stupid by people like you and they
I think a lot of them
believe.
are actually. I'm not going to lie. I mean, I do think same with the flat earthers. I think a lot of flat earthers. I mean, every
I
flat
think a
earther
lot
I've
of
met
them-
is dumb. Everyone. I can't say that with the younger creationists. I've actually met a lot of intelligent. You're a very intelligent guy. There's no question about that.
Well, thank you.
Yeah,
I
no question
appreciate
about that. There's a distinction there. I don't know if you ever talked to flat earthers, but I cannot think of a single flat earther that I said, "Damn, this guy." You know,
I was not aware there were any still around or
it's
ever
huge
around.
around here. Are you kidding me? Matter of fact, if I tell you what, if I wanted to change my channel to flat Earth, I'd be far more popular. I'm not even kidding.
They say that there are flat earthers all over the globe.
Yeah,
I love that expression.
Yeah, well,
but I'm not even lying, guys. Tell them that I'm telling the truth. If I
believe
change this like flat Earth debates, how many more viewers would I get? It would be pretty. Yeah.
Well, it's definitely an a hot commodity. Certainly, just for Ben's benefit. I mean, there've always been flat earthers around, but it's almost like a fad has really taken off. I would say about the past 2 years, very strongly in the past year. There have been some people who come forward. Just a lot more than ever, really. Certainly since the history of this sort of gathering on the internet. I will say, and I'll connect this to the point that I think you were trying to make, and I hope you go ahead and make it. But they're certainly subject to mockery. But I'll tell you, there's one thing I noticed about the flat earthers. I'm yet to find a flat earther that remembers like SOHCAHTOA. Right, is able to take a triangle. Like they'll come in and they'll say, "Well, we can do measurements. You can prove to yourself that the Earth is flat. You can do experiments to prove the Earth is flat." And yet, if you get into a discussion with them about anything, about measurement, about optics, about coordinates, about just something like, well, if you have a triangle that's got one right angle and one 45-degree angle, what's the other angle going to be?
Depends on the geometry of space.
They'll boot the ball.
Euclid's fifth postulate.
I'm just talking about basic high school math. They'll boot the ball on it, and I can tell you in my experience with a lot of discussions with young Earth creationists, I find that's kind of the same phenomenon. Is that they'll be very passionate about their beliefs, but they really won't have the basic toolbox that a person would use to go about finding out about acquiring true beliefs.
Well, I'm not a professional. I'm not a professional scientist. That's not what I do. But part of the reason I don't do it is because of the discrimination that exists in the academy. And I know that if I tried, I would either A, have to hide what I believe, period, and not reveal it, or B, I would say what I believe and risk my career. That is
there's an old adage.
That sounds like
what?
That sounds like sour grapes. This is something. This again.
It's not sour grapes. It's based on multiple. I had a guy who I knew personally at UNL, who was an astronomy professor there, and he signed a little document that made him the faculty advisor for a club that I had, an intelligent design club. He was denied a position at the University of Kentucky to run a new astronomical observatory there. This is some years after I knew him. And he actually won a court case that was alleging discrimination because they found that as part of the court, he came up evidence, emails from faculty members discussing his hiring, saying unequivocally that he was by far, that's a direct quote, by far the most qualified applicant. The reason they did not accept his application was because he was, and another quote, potentially evangelical. And also that
and those things do happen, and I'm not advocating this.
And they would happen in lesser degrees to any young Earth creationists who participated in academia.
I have to tell you, there's a rule of
Can I just say
thumb.
in his defense, if I were ever helping out with the interview of a candidate, and they started going off on their young Earth creationist views, I'd be hesitant to recommend them.
Yeah, and I agree with that. And I'm going to support that in a second here. But I will say that the rule of thumb is when you enter the laboratory, your beliefs stay at the door, right? Your religious beliefs, whatever you believe, it doesn't matter. When you're in the lab, you're in the lab. That's an old adage with that. But as Herman points out in the live feed, he says, he says, quote, "We scientists discriminate against people who are not doing science," and that's where I'm going back to the fact that a creationist, young Earth creationist, can believe what they want. They have the freedom to do that.
That's not why you asked me if I would
but-
change my mind.
But
though.
they're not doing science at that point. If they have a
Then
confirmation
why
already,
did you care?
but it is a fundamental problem in science that you cannot have a confirmation ahead of your data. You can't do that. That's not how science is
That's
run.
why
So
there's peer review.
that's
And
not just what peer
criticizing
review is
each
for.
other.
You cannot start off with a confirmation bias to saying doing science, and that's why they're excluded, because they have that position. They're starting off with a conclusion. That's why they're excluded. So it's a principle of the fact they don't do scientific methodology correctly.
Scientists have to start out with conclusions, and they say they
No,
get
they
them
don't.
from other scientists that come before, to which they have not investigated those claims themselves, and sometimes they're wrong. Historically, we know that.
But if anybody can demonstrate they're wrong, then that's great, right? I mean, things
That's
are up
great.
for review all the time.
But people don't change their minds.
Sure, they do.
Oh no, that's
Wait,
the whole point
wait
that
a
Thomas
minute.
Kuhn was making.
Wait a minute.
And it's,
Wait a
and
minute. Did you just say that scientists start with a conclusion?
I
Yeah,
say
I don't know
they
how that would
start
work.
Did I
with
misunderstand?
they start with prior conclusions that they base their work on. There's a famous quote from Einstein: "Standing on the shoulders of giants." It's part of the science. You can't take it out. You have
No,
to
but when
stand on the
right,
shoulders
no, but
of
you
giants.
understand when they're developing their own hypothesis, right?
That's
they don't
Newton,
start with
not
that.
Einstein.
Okay, this Newton, whatever.
But you don't start off with the hypothesis saying I've already determined my hypothesis to be correct. That's what I'm saying by starting off by
You
conclusion.
don't do the experiment if you don't believe that there's something to find out.
But you don't do the experiment you don't do your experimentation to support your hypothesis. You do your experiment to falsify your hypothesis.
That's not how you get papers published. If that were correct, there would be 90% of the papers that are published would be falsifying results. But you don't see that, do you?
no.
Wait. How
That's not-
can a paper be 90%? Where are you getting this from? 90%
I'm
of
saying
papers.
that
will be-
most, if that's really what
You
scientists
get papers published by failing to falsify things.
Right, there you go. You fail to
You set out-
yeah, exactly. You
set out to attempt to falsify, and
Right.
then you fail to falsify, and that's the
If
interesting.
you can't
thing
reject the
that
null hypothesis,
But
then you
you
accept
should
the claim of
report that.
hypothesis.
because other people might have that hypothesis. And if you report the failure, the rejection, or the falsification of the hypothesis, that's important to do as science, and it ought to be published, but it's not.
That's how science
Not
is
often.
progressed, through falsification.
Right, but I mean, it's important for people who want to say, "Look, I've already done this and it was falsified. Let's come up with a new hypothesis."
or
So
the
other
attempt
people
of
don't waste time with it. But when you publish a paper, you're basically saying, "Hey, look, we ran all these tests to try to show this to be wrong, and we were able to reject that null hypothesis within a certain degree of accuracy. Like 0.05 is your p-value usually, but it could be depend." And you can say at that point, we determine there's a high confidence level that this is a correct hypothesis.
So why don't they publish papers when they actually do falsify hypotheses?
Well, why would you bother?
That's why.
Because it's a community,
things-
and other people
No,
might
no,
have
no.
the same wrong
That's why
idea.
I mean, if I have okay, look at
Right.
it. But if I have a hypothesis that little green men cause gravity to work,
What?
and I run all these experiments and if it doesn't work, why would I have a published paper saying, "Look, little green men don't cause gravity"? I mean, you don't publish papers to show people what you were wrong about. You publish a paper to show what you think you were right about because you couldn't falsify it. But I mean, there are articles you may publish, and there might be information out there to say, "Oh yeah, we did try this; it didn't work." So other people don't waste time doing it again and having the same
Exactly.
hypothesis. But
That's
people
you
do
just
that.
making
I think
my
they do write
argument
things
for
like
them.
that.
Well, it's like a good example of that would be, previously we didn't think that, and I might be wrong. I always get this wrong. Soft tissue in that bone.
What about it?
I'm a layman about it. Anyway, we previously thought that kind of thing couldn't happen, but now you publish a paper saying that we were wrong.
Yeah, the
that's-
rule of thumb was that they couldn't find
Because
any
they
kind of
found
protein.
soft tissue, not because
Well,
they
it wasn't
changed.
really technically soft tissue.
Yeah,
I
I
find
know it was, and I just
I
I
mean,
don't know.
you can't really. I can't call something soft tissue when it was hard as a rock and had to be soaked in acid just to get some collagen out of it.
I know what it was, but I don't remember the technical term.
It was collagen and heme byproduct. Heme
Okay.
compounds. But see, but the science began when Mary Schweitzer said, "Oh, look, I need to come up with a way to explain this and see if I can find some hypothesis." And she actually did some experimentations. She developed a mechanism that she had tested over a period of 2 years with ostrich bone, and it had to do with the iron as a preservation mechanism. She had an independent study done by independent laboratories that said, "Yeah, this actually works." That's how science is done.
Hey, I gotta say my goodbyes. I do want to say, Ben, it's nice to meet you. It sounds to me like there was a good discussion. I was doing other stuff.
It was. No, it is. I'm actually enjoying this. I'm actually really.
Well, just let you know, I do want to go back and listen to the opening moments, the hangout, and kind of see how the discussion about human chromosome 2 went. And I do wish I could have either participated in or at least listened live while it happened. I will listen to it subsequently. And I do have a couple of questions and discussion topics for you, but I have to disassemble my computer and take the hard drives out, and I don't think I can stay in the hangout. Does anybody know about
when you do that? When you take the hard drive out?
Yeah, I did not. Yeah,
it's not hard drive out.
Yeah, no.
I will probably lose my connection.
Yeah.
Okay.
Well, I mean, you when you watch, I tell you what, when you watch the beginning of the hangouts like that. I mean, I'm sure Ben is part of the creationism on Facebook. A lot of us from Great Debate Community are in there. So, if you have a follow up later on, I'm sure Ben will be happy to join us again. He's more than welcome to.
Yeah.
So.
And
okay.
I'm planning on making a couple more posts about a couple more evidences of the young Earth. I already posted on the helium diffusion one a few weeks ago.
What do you think of Paul, by the way?
Ben and Steve, what I'd appreciate. Oh, I'm sorry. Steve was talking.
No, go ahead. Finish up.
Could you, if Ben's got, actually, sorry, I just see his YouTube channel here. Okay. Where do you do most of your posting, Ben? Is it a lot on YouTube channel or elsewhere? I'm not a Facebook guy, so don't talk me into it.
I don't use my YouTube channel. I just go on the Facebook.
Hey, can we do this? How about we do this? Now that you have a Gmail thing, you've been added to the Great Debate community, right?
I don't know. This is the first time I've been on.
You know what? Let me make sure that you are. And what you're going to do is just whatever you post to Facebook, either pop it on the Great Debate community, or I can tag it on it as well. But
yeah, because I'm hearing something. I mean, like I say, I get a little frustrated with the conversation because I hear a lot of the same things over and over again. And there are some things you said that kind of made my ears perk up. And I apologize. I'm having this problem with my computer. I have to take it apart because it seems I kind of feel like I'm missing an opportunity. But hopefully you or Steve and you together will provide other opportunities where we can chat. So
yeah, I mean, he's actually arranging for hopefully Tomkins to actually join us in January. So that's not that far away. It's another month or so. So I'm sure Tomkins wants to watch this video. He may or may not have time. But Ben was saying that Tomkins may be willing to join us in January. And I think that obviously, Hagda, you'd like to be there for that.
Yeah, I would like that. I mean, we'll have to sort of pick. Again, I don't want to sort of inundate the guy too much. And I know when I had been in contact with his office before, well, he may have been really busy at the time. Well, we'll talk about it else.
Well, yeah, but Ben's a creationist, and so he kind of does talk to them more than he does like with Riff or you or somebody.
Okay, sure enough.
Yeah.
All right. Well, nonetheless, I got to get some surgery going here, so we'll chat with you guys later.
All right, thank you, bud. I put in the group chat there the Great Debate Community link. Just click on that, Ben. Ask to join, and I'll add you. And then if you can post whatever you post in the Creationism forum, post in here as well. This is a kind of a get-together site that has like all sides that are, so it's not. It's a little more stringent than the Creationism one because this is actually the posts have to be relative. They have to be on topic. They have to be something that people can talk about, right? It's not for like a spamming.
You have all those debate group rules. I've been in groups like that.
Yeah, similar things. There's a little more structure to it. That's all.
You have to respond to questions or something. Is that one of the rules?
No. I know if you post something, you don't have to respond. It's not like the creationism versus evolution one that I know you're talking about. That if you post something, you've got to be available to respond to it, right?
I got banned from that one.
Yeah, I don't know if that's really a great rule. I understand it, but I mean, not everybody has the time to do that. But I think maybe in 24 hours, I think you should be able to respond. But
they were just for a while when the first post I made, they started spamming me with ridiculous questions, and then told me I had to respond.
What do you think of Paul? If I can ask, we've had Paul on, Paul Humber.
Oh, the guy who posts all those memes?
Yeah, the CR Ministry ones.
I don't read those.
Yeah, he came on. He didn't. I mean, I'll tell you right now, we disagree with you violently, but I mean, you did know the topic far better than Paul did. Paul did not know anything. I mean, just nothing. It was really kind of embarrassing.
And that's the part that I was getting to with the cultural thing is these guys, we as creationists feel that we're excluded, and we don't have people who, like Dr. Tomkins, are allowed to participate fairly in the discussion. And so people like Paul Humber, and even myself to some extent, we feel defensive, and we feel like we have to defend it when we don't necessarily have all the kind of background knowledge that would be.
Well, we've tried to get in touch with them. I mean, we've had, obviously Fuzz Rana join us, and we had Dr. Ross join us, and we're trying to.
Ruhif contacted him, and he directly addressed Ruhif's criticism in a paper about the bug in the BLAST-N program. So if you want to, you can talk to them. All I did was I called ICR. Obviously, they're not going to give out.
No, but we were told that he was too
information.
busy.
He is extremely busy, is what he stuff.
Now
that
I
he told
talked to Kevin Anderson. I've talked to him in email. He just outright doesn't really want to defend Tompkins' work, is basically what he said.
Well, there's disagreements in the creation science community, just like a real science community where they have disagreements and they try to prove each other wrong sometimes. And some theories get falsified and left behind, like the vapor canopy theory, and just about everything from Kent
Hydroplate field theory.
Everything from Kent Hovind you've ever heard.
How can that be?
Well,
how can that be when places like ICR and Answers in Genesis make their, in quote, scientists find a paper that says they even with if evidence contradicts what they're trying to push, they're going to reject that evidence. How can you say that they try to prove each other wrong when that's
you guys are trying to prove that we're wrong about something that is foundational to creation science. So if we were wrong about it, if we were wrong about it and believed we were wrong,
as creation science. So
if we were wrong about it and believed we were wrong about it, we wouldn't be creation scientists anymore. So it's kind of.
Do you know what Tompkins does nowadays? I mean, do you know what he's busy at? What do you? I mean, just out of curiosity.
No, I don't know the guy. I called him once, and I talked to him for about an hour and a half, and he seemed interested. And he's definitely still knowledgeable about the issues. He rattled them off to me, so he is thinking about them. The specific ones, a lot of them that Ruhif raised, he knew about him, obviously. Ruhif's contacted a lot of them.
Yeah. Obviously, Ruhif hasn't been feeling well. He asked me if he wants to come up with a bunch of questions to ask Tompkins if he's not available to be actually in the hangout.
Well, I would suggest to him look at Tompkins' paper from 2015. So that's where he addresses the bug in BLAST that Ruhif found. Which, by the way, if Ruhif hasn't published that, he probably should talk to other people about it too, because that's kind of an important.
Well, it's not really to publish. I mean, his reputation. I mean,
a lot of people. No, I'm talking about the bug in the program.
That's long been. Yeah, that's long been established. They've already fixed all that.
Okay. Well, that's good.
Yeah. But anyways, do you guys want to add to this? People are asking if I want to have a after hangout. Probably not. I think we've kind of gone long enough. We obviously we can have another hangout multiple different times, but I kind of wrap this up. So I'm going to give everybody the last word, and then Ben, I'm going to give you the last word, and me. So let me start real quick, going from across the board here. Rational, do you have anything to add? No, he's already fallen asleep. Mike. By the way, Mike used to be a younger creationist, so I mean, he might be a person to actually talk to about how somebody actually changes from a younger creationist position to a non-younger creationist position. Because I mean, it is a train of thought, right? I mean, it is a mental thing, correct, Mike?
Well, It's a mental thing. What do you mean?
Well, I mean, because when you're so indoctrinated in the creationism, I mean, you have to believe it, you have to accept it, or then you're kind of outcast if you're not. I mean, the young creationists taught that
Yeah,
it cannot be wrong, right?
It's a cult. No, it's all it is a cult. So it's a. I think it's a matter of being honest with yourself.
So I'm a cult member.
Yes.
Okay.
Isaiah, did you want to add something?
Hey, Mike, how are you doing?
Isaiah, you there? No. CRISPR, you want to add something? Okay. CRISPR, you there? Okay, go ahead.
Nothing really occurs.
Okay.
No.
Before I give Ben the last word, I wanted to ask Ben one last thing because you said something and I kind of like glazed over. But you're not a big fan of Hovind?
No.
Okay.
I mean, I heard all his stuff, like all the other creationists did. Well, other lay creationists did. He was a popularizer of science.
Yeah, but he made a lot. I mean, every one of his claims is trivial to debunk. I mean, he has so many bad claims, and I think that shows, like, from young creationism, that they a lot of them got their start from guys like him. And
there's a lot of, I don't believe that. There's a lot of hucksters out there that prey on people who feel defensive, and part of that is constantly being derided by other people, and so they look around desperately looking for something to defend their position, and they latch on to guys like Hovind or something like that.
So you don't think he's valid in any way, or you think he's a con man?
Well, he was certainly making money off of it. I don't.
You know what he's trying to push now? Do you know his two big pushes?
I thought that he was pushing to stay out of jail for
tax
evasion.
That probably no. He's pushing anti-vaxxing, and he's pushing Laetrile as a cure for cancer.
He's one of the anti-vaccine people.
Oh, big time now. He married an anti-vaxxer.
Yeah, his wife is a
Tito. What's her last name?
Anti-vax advocate.
Yeah, I mean, in fact, I just posted something on there the other day, and I had some people already start up with me, but I was like, this is just complete. Oh my!
Yeah, Mary
Toko.
Toko, whatever. Yeah,
and I mean, it. I've talked. We've all talked to Hovind, and we've all talked to Eric many times here. Isaiah's had 2 debates with Eric. When I see Hovind and Mary together, is it just me, or is there something kind of creepy there? I'm not trying to be mean. I'm not trying to be disparaging, but there's something just to me does not sit well. Am I the only one that thinks this? Is this my imagination?
What are you talking about? The between
whenever they do a video together, it's just I don't know. It's just weird.
Like it's being forced.
Yeah, it's just I don't even know how to explain it. It's just I don't know if he even buys into this anti-vaccine. I think he just did it for her.
I'm picking up on the same thing you are.
Yeah. But anyways, we can have a discussion with Hovind another time. I don't know if you ever ran into Hovind. He's actually okay to talk to. I mean, it's not like he's like a horrible person to talk to. It's just that he just gets everything wrong. It's so amazing.
Okay. And I know that because you like to make fun of creationists, you like to focus on people like Hovind. But
they're soft targets.
If you're being an intellectually honest person, you want to focus on the strongest argument of the person.
Agreed. Yes. Yeah, absolutely. Hovind's easy. We've migrated from Hovind. We've already, like I said, I've taken on Jay Hall, who was the author of Younger Science. He's been with us. We've taken on Wayne Fillmore. We took on, and he's no longer a Younger Creationist. He actually has given it up after having a debate with me on macroevolution, and then with General being explained to a lot of things. He actually is one of the people that actually no longer Younger Creationists. There's a handful in the community that have gotten out of it.
Can I say
they are soft targets, yeah. I'm not going to lie. We have some easy people, sure.
Ben, you said that you put no stock in what Kent says. Hold on. Okay. When I say that Younger Creationism is a cult, I'm talking about Hovind's side, and you said that you don't put a lot of stock in what he says. So I take that back. I think you're wrong still, but I take back the whole cult thing.
Okay.
I'm sorry.
Thank you.
Yeah, I mean, I'm not going to place
Thanks.
like Dr. Tompkins or Humphrey into the cult label. I want to do that, but I'm going to place anybody who follows Hovind into it because I do look at him as a cult leader. He acts like a cult leader, right? I mean, he's not just putting out information on AIG. He's actually he has people literally following him and donating money to his ministry.
He has a compound now. So
Yeah, I mean, to me, it is very cult.
it's Hoventown. It's gonna be Kentstown,
Yeah.
Kentville,
Well,
Jonestown.
When I was looking through, I think it was on the Larry Moran blog post, Tomkins told me he thought that's where all this kind of blew up, and suddenly everyone's interested in this issue, the human chromosome fusion thing.
Well, like you said, I mean,
Is-
you want to get the bigger arguments, and you want to go against. That's why we want Tompkins, and we want those other guys, Humphreys or anybody. Perdome. I've talked to Dr. Jonathan Sarfati.
Yeah, I've heard
Yeah,
that.
I mean, I've talked to him numerous times, and I've asked him to come in, but he's always reluctant to. But I mean, we have wanted to get them because I mean, going against Hovind was trivial. I mean, it was not a challenge. I'm sorry, it just wasn't.
Oh, I was meaning to read actually a quote from C.S. Lewis. That's my avatar. I just picked it. But he said something about the minds you daily meet are just like yours, and you should, basically engage diverse opinions. That's part of the reason why I'm here.
I like C.S. Lewis. I like a lot of his writings, actually.
That's who that is in your avatar.
Yeah, I don't.
I knew that's who it was. Okay, confirmed. All right.
But what I wanted to say earlier was that in that thread there was Ace of Spades, Diogenes, and also Glenn Ruheef were talking together, and basically they said Tompkins obviously knows his stuff. He's
Right.
way smart enough to know his stuff, and so he accused Tompkins of not being ignorant but being a liar. And that's the sort of thing that makes people like Tompkins not want to participate in a debate or in a forum like this, because, well, obvious.
No, I can see that, and I do admit that. I don't want to call people liars like Tompkins. I mean, I think what it was, because he is somebody that does know his material, right? I mean, he's not the fruit on the ground, and I will tell you, you knew the material a lot better than like with Paul is like that, right? I mean, obviously you are someone that has at least read up on some of these things, okay? So you're not coming in blind. But Tompkins, when he makes these kind of these statements, I think Ruth is either saying, look, either you're lying or you're just being incredibly inept in this one area. Why? When you're so good at all these other areas, right? I mean, even Sam says he's looked at his research on botany and genetic sequencing of prep work or whatever he was doing. And he said he actually was pretty competent in his field. So,
And he would only know that because he's coming after him because he's a creationist.
Well, because when people quote these people, right? When we say, okay, what's your evidence? Okay, this is a typical situation we'll have. Like, for example, we'll have somebody come in and say, oh, well, it wasn't a fusion. Okay, why isn't it a fusion? All they will do is quote Tompkins rather than trying to explain it. You attempted to explain it, which is far greater than what we've had in the past, right? Do we agree with you? No, but you've at least attempted to explain it. We don't usually get that. We usually have people coming in that says, oh, well, here, like Paul. Paul didn't attempt to explain Tompkins' work. He didn't understand it. He just parroted Tompkins. I'm like, well, anybody can do that, right? That's not what we're looking for.
I understand that, and I try to do the best I can, but understand that it would be much better to have Tompkins in here.
Sure.
But
sure. And hopefully, you can talk to him and say, look, you did your best to try to explain his work, and do you think that we are a group of people that should be worthy enough for him to come in and talk to? I mean, you obviously realize that the amount of intellect that was in this one room was immense. I'm not gonna lie, we had very smart people. I mean, half the people in here were masters, PhD students. I mean, they're not your amateur kind of run of the mill person. You know a lot of them. So. I mean, he's not like he's going up against people that just are idiots. So if he really wants to
say that,
so if he really wants to like explain it to people, we want his explanation, right? We want him to explain his methods because he didn't have a method section. Does that make sense?
Yeah, that's fine.
Cool. All right, I'm going to give you the last word, and I'm going to do my 30-second blurb that I do with any hangout. So Ben, go ahead, take it away. You have like a full minute of your own. Say anything you want, because I want to make sure that you definitely get the idea that you have not been censored or limited in your speech or anything like that. So say what you got to say, and let me know when you're done.
I appreciate the sentiment. I don't have anything to add at this time.
Did you enjoy it?
Yeah,
Good,
I
good.
always do. So thanks for having me on, and I'd love to hang out with you guys more often.
Yeah, no, you know what? And we don't, when we do, we just have regular hangouts too. So you're more than welcome to come by during regular hangouts. We talk about all kinds of different things. So, I mean, we have an eclectic
Sure.
group. So it's not just about, like theology or run with creationism, we talked about Pizzagate the other day, for Pete's sake.
I don't even know what that is. I saw
Yeah,
it
it's
on
not
your-
worth knowing about. I'm sorry. I'm going to say this on air again. I don't think there's anything to Pizzagate, guys. I'm sorry, Mark Anoia. I love you. I think the word of you. I think you're good at what you do. I cannot agree with you on this one. I think there's nothing to Pizzagate, and I'm sorry. I feel bad because Mark and Mark and Nevin and those guys try to put forth a good argument. I just can't see it.
What's Pizzagate?
You know what? Watch my videos. The most popular term on Google right now is hashtag Pizzagate.
Okay.
It's about reptilians eating pizza.
All right, I got to wrap this up. People are getting antsy. Thank you guys for watching as usual. This was a very good special hangout. I am going to be trying running hangouts pretty much Monday through Friday if I can, at all possibility, 2 o'clock to 3 o'clock to as many hours ago. So depending on when I want to start, be 2 or 3. But I'm trying to run hangouts more frequently than I have been. But anyways, my links for the hangout that everybody had in the group chat here, I put most of them, thanks to Isaiah, into the video description. So if you want to kind of follow along what people were looking at, it'll be in the video description. I also put the links for Patreon. Again, I do these channels to support myself. So basically, if you want to help support the channel and donate, anything is completely appreciated, nominal or not. It means a lot, and I do give back to my patrons. We're trying to get some bigger names in here, and I do make videos. So anyways, guys, thank you very much. Ben, thank you. You were an awesome guest, and I really want to thank my panel and everybody else who joined us tonight, and those who are watching, and for those who will watch. Okay, thank you guys.